So welcome everyone to the Open Neurodata Showcase webinar today. We have a great afternoon event. We are going to be recording this webinar, just FYI, and we also will be able to answer anyone's questions in the Q&A panel. I will be moderating those if people want to submit questions along the way. Ben is going to start us off with an introduction for the day. Ben, do you want to take it over?
Yeah, thanks, Carly. All right, hi everyone, and welcome to the Open Neurodata Showcase. This is a collaboration between a number of different groups. So the Berkeley Lab, the Kavli Foundation, the DANDI group at MIT, Neurodata Without Borders, the Allen Institute, and Catalyst Neuro. And I'm Ben Dichter, the founder of Catalyst Neuro. So welcome to the first Open Neurodata Showcase. So just to explain kind of the goals here. So the Brain Initiative has supportive software for sharing neurophysiology data in a way that promotes reanalysis. So this includes data standards such as Neurodata Without Borders, which helps us store neurophysiology in a way that packages all of the data with necessary metadata for reanalysis. Including the DANDI archive, which is an archive where you can deposit such data, and it can handle data on the scale of terabytes, and you can search and download and reuse data. And then projects such as the OpenScope program from the Allen Institute, which produces really high value data sets and devotes the necessary attention to packaging this data in a way that many labs can use it. And the idea behind this OpenScope program is to create data sets that the community at large can use. And they leverage the Neurodata Without Borders standard and the DANDI archive to disseminate this data. So we're going to have introductory talks for each of these topics. But this is really just kind of the framework of an infrastructure that enables reuse in on a scale that really hasn't been possible before for neurophysiology. And using that infrastructure, there are now over 100 DANDI sets in the Neurodata Without Borders format on the DANDI archive, including really high value data sets from many groups, including the OpenScope program. So now that this infrastructure is in place, and of course, we're still working on making that easier and more efficient, et cetera, better searching, all of that. But while this infrastructure is in place and we have over 100 DANDI sets, now we can start to shift our focus into actually helping neuroscientists use these resources to generate new scientific insights. So, so we're really focusing now with this event on how can we facilitate these interactions between the data contributors and the people interested in reusing the data, so that we can really get this reuse cycle to flourish. So the way I think about this is that our ultimate vision is right now when a scientist has a new question, a neuroscientist has a new question, they think, what experiment should I design and run to start answering this question? I would like to, in some cases, change that to what query should I run on existing data, which would allow me to do a pilot analysis and move to the next step using existing data as opposed to always collecting new data. But that is a really big task, actually. It takes not only a lot of technology in standardizing and disseminating data and collecting obviously high value data sets, it also takes a societal shift in the field. And so I'm really excited to see now 56 and growing participants here, and because we really need your help with this. And we know this is going to be an iterative process. This is our first event trying to do this, and we really wanted to make it open to anybody and virtual so that anybody in the world could participate. And what we're really hoping for here is to try to make an attempt to connect the data contributors to those interested in using this data, and identify possibilities for collaboration, and also potentially identify areas where we need to enhance the ecosystem to facilitate the most efficient collaboration. So, so I'm really excited to see all of you here, and I really think this is a huge step forward in our vision of being able to reuse neuroscience data. So, walking you through the agenda for today, Right now, we are here in the introduction, and then we're going to have introductory talks, one for NWB, Neurodata Without Borders, given by Oliver Rubel, one for DANDI, the archive, given by Yara Kalchenko, and one by Jerome Lecoq for the OpenScope program, which is disseminating neurophysiology data. Then we're going to have a little break, and when we come back, we're going to have a roundtable discussion where we talk about the pros and cons of reusing data sets, questions to ask, caveats, and we have a really great panel here with Bing Brunton, Mackenzie Mathis, and Colleen Gillen. This will be moderated by Saskia De Vries. Then we're going to have a data blitz. So we have 12 of some of the most high impact, we think, really impressive data sets on DANDI in NWB. We have the contributors of those data sets have prepared a data blitz slide. So each of them are going to get 1 or 2 minutes to talk about their project. So we're going to kind of go rapid fire through 12 different projects. And then we're all going to move over to Gather. And if you've never been on Gather, it's basically like if you've ever played Pokemon on Game Boy, you have like a little 2D environment, and then you can walk around, and when you're near someone, you can talk to them. So we're going to go into the Gather virtual space and do a poster session within there. So really the idea of the data blitz is just to give you a taste, just a teaser of all of the different data sets. And then you will have an hour and a half to go in and have detailed conversations with the contributors of this data and really figure out the best way to reuse it. Also, I want to mention this event, it is a standalone event, but it also doubles as day 0 for Neurodata Rehack, which is an upcoming event at the Allen Institute next week, where we're going to meet with people in person and hack. So whether you're part of that event or not, we welcome you here. We really wanted this particular first day or 0th day to be open to everyone. So this is a snapshot of all of the data presenters you're going to see in the data blitz and in the poster session. I'm really happy about the diversity of this. I mean, we have different modalities, including electrophysiology, optical physiology, FNIRS, different functions of the brain, including sensation and motor control, including navigation and memory, different species, including mice and rats and non-human primates and humans. So this really showcases the diversity of the data available in the DANDI archive. And we really found high value data sets all across the spectrum. So I think that's really going to be great. So I just want to kind of, to motivate this, I want to talk about the different ways that you can reuse neurophysiology data just to get you thinking as you go into these talks. So the first one is the way that I've already described, which is you run a pilot analysis on existing data before you design your own experiment. But there are a number of other ways you could interact with existing data. You could also say you have a project, you already have a result from your lab, and you want to see if this result is consistent with other species or brain areas. This is a really great way to increase the impact factor of your publication. You could also showcase the capabilities of your tools or analysis on existing data from another lab, or you can build new tools or analyses or pre-processing programs against shared data. You can explore follow-up questions to a study. I think this is really great. I mean, even if you don't have a specific question going into this, you can just talk to the people who collected the data. And I guarantee you, many of them are going to have outstanding questions that they would be happy if someone else explored, particularly if people are going from academia to industry. They'll have a number of unexplored questions that they won't really have time to answer. So this is a great opportunity for you to kind of pick up where they left off, and maybe they'll help you do that. Another really great option is doing an aggregative analysis. So now that all this data is standardized in a common format, you can aggregate across these data sets and gain statistical power that you wouldn't be able to get from a single lab. So I think that's a really cool new opportunity that is enabled here. And finally, I just wanted to kind of expand this to, you can use this data even outside of the traditional academic flow of new scientific discovery, you can actually just use this as an educational resource in one of your courses. I think that would be a great way to use this data and help the next generation of scientists really learn what real data looks like and how to do real data analysis using data shared in this way. So I just want to thank all of the organizers. This is a huge team effort. We have Oliver Rubel and Ryan Lee from the Lawrence Berkeley National Lab. We have Satra and Yarek from the DANDI project at MIT. We have Jerome, Carly, and Caitlin from the Allen Institute, who really did a lot of behind the scenes stuff, creating the website and creating this webinar and emailing everyone, et cetera. And we have Stephanie Albin from the Kavli Foundation. So this has been a big team with a big team effort with a lot of different people interested in promoting data reuse. So with that, I want to pass it on to Oliver, who will give a talk on Introduction to Neurodata Without Borders. And I want to thank you all for being here with this first Open Neurodata Showcase event.
And just, I'm going to plug a reminder before Oliver takes over with the slides. If you guys have any questions during the talks, you can add them to the Q&A box and we'll moderate them and then be able to answer those after Oliver's talk. And just another reminder, the session is being recorded. All right, Oliver.
Well, thank you very much, Carly and Ben. Welcome everyone to the Neurodata Showcase. I'm really excited to see you all here, and I hope you will have a great day. My name is Oliver Rubel. I'm a staff scientist in the Computational Biosciences group at Lawrence Berkeley National Lab, and I'm the lead PI together with Ben on an NIH U24 grant that currently funds the development of NWB, and I've been involved with the development of NWB in the leading role since late 2016. And so in this talk, I just want to give you a very quick overview of NWB and the larger NWB ecosystem. I'm not going to go into details. We have lots of online tutorials and recordings from previous user training events if you want to learn more about the details. But here I just want to give you a really quick introduction, in particular for those folks that may be new to NWB as you enter this event. Sorry. And so many of the key challenges we face in NWB and neurophysiology more broadly are really driven by the diversity of experiments and methods. The neurophysiology of cells and tissues are monitored across brain regions in flies, rodents, humans, and many other species. We are optical physiology, extracellular electrophysiology, intracellular recordings through simulations, and within each of these modalities through a broad range of different devices and methodologies. And then in addition, we also have to monitor the behavior of our subjects. We have to describe the design and of the experiments, monitor execution of experiments, and also describe the environment. And so this in turn leads us to really diverse collections of multimodal signals and data processing and analysis data products that we need to collect and describe even just for single experiments. And so understanding the brain requires integrating of all that data across the diversity of these modalities and products that we're acquiring. And so the data must be findable, accessible, interoperable, and reusable to really make the most use of the data. And so it is this diversity of experiments, data types, application uses, and stakeholders, and really the rate of methodological innovation that are the key challenges that we need to address to advance standardization of neurophysiology data. And so as a result, some of the key challenges that we face in neurophysiology today are that neurophysiology data are expensive to collect. Sharing of data within a lab and with collaborators is hard, and scientific results and analysis are hard to reproduce and compare. However, these challenges on the flip side also provide us with great opportunities in that neuroscience data sets are often very rich and can be used to answer many different questions, even years after acquisition. And at the same time, reusing data saves money, it saves time, and it reduces the need for animal use. And by providing efficient methods for standardizing neurophysiology data with NWB, we can facilitate sharing of data and collaboration within teams and across the community, and also enable consistent definition of data, integration and sharing of analysis tools and methods, which in turn facilitates the reuse of data and reproduction of derived conclusions. And so the creation of NWB has really been motivated by these challenges and opportunities. And so NWB, first of all, defines a unified data standard for neurophysiology data that is focused on the dynamics of groups of neurons measured under a large range of experimental conditions. And so NWB supports a broad range of data types from end-to-end neurophysiology experiments, which includes recordings of neural activity via extracellular and intercellular electrophysiology, as well as optical physiology, behavior data, as well as metadata about the data acquisition, such as devices, experimental design, such as stimuli and trial structure, and experimental subjects. And then NWB, importantly, also supports the formal extension of the data standard to enable the community to add new data types and modalities, as well as metadata that's specific to the labs, the acquisition, processing, or the experiment. And so NWB, in that way, enables unified storage of multimodal raw and processed data from neurophysiology experiments. And so just as an example here, you can see a pipeline where the experiment consists of both extracellular electrophysiology and optical physiology recordings, which are measured simultaneously. And so using NWB, the scientists can then store both the raw optical and electrical recordings, as well as subsequent primary data pre-processing results from spike sorting and RI detection in a single NWB file and share that file with collaborators. And so with this, NWB provides a critical conduit to facilitate analysis of the complete data from neurophysiology experiments. So for example, at the bottom part of the slide, you see an interactive dashboard. On the left shows the acquired image from the optical physiology with an outline of a segment of a single neuron. And on the right, we can then see a juxtaposition of the extracellular electrical physiology recording, the extracted spike times, and the simultaneous fluorescence recording of the segmented region, so we can analyze and study that data in context. And so one of the key challenges in the development of experimental data standards like NWB is, as I mentioned before, really the diversity of the stakeholders and the data and the requirements that we face throughout the data lifecycle, from acquisition of the data to analysis and publication. And so a key aspect that has allowed us to manage these diverse requirements and really the discussions in the community is that we already very early on outlined and defined what the main components of the NWB standard are, and that we need to address as we develop NWB. And so those components are roughly outlined here in the triangle that you see on the slides. And so first we have the NWB specification language, which allows us to formally define neuroscience data standards and model the data. And there's the data standard schema, which is shown in orange, which uses the specification language to organize complex collections of neuroscience data. And so this is probably the component that most of you will think of as the formal standard. It's the description that governs how the data needs to be organized in NWB files. Then there's the data storage component, where we look at how can we most efficiently take all these collections of data and to store them efficiently on complex storage devices, whether that is in the cloud, a high-performance compute system, or local storage. And then finally, there are the data APIs, which target efficient interaction with the neuroscience data to read and write, query, and analyze NWB data. And so in this talk, I'm not going to go in detail through all the components of the NWB ecosystem. You will be able to find more detailed introductions to these separate parts, as well as a more detailed overview talk on our YouTube channel, as well as in our online documentation. However, for those of you who may be new to NWB, I just want to give you a general idea of some of the basic concepts we use in NWB to organize neurophysiology data. And so we'll have a quick look now at a high level organization of extracellular neurophysiology data in NWB to give you an idea of some of the basic concepts. And so first of all, data is organized in NWB hierarchically via groups, which you can think of as folders on your file system. So the organization is in that way familiar in that, if you know how you organize data on a file system, it looks similar inside an NWB file. And then we define a broad collection of reusable, what we call neurodata types, which you can think of as different types of data that you can use to store broad collections of data. So for example, we have a base time series that describes how we store time series recordings, and then there's more specialized types of those time series, such as electrical series for storing extracellular recordings, or 2-photon series to store OFIS recordings. And so at the first level, we then have general metadata about our experiments, which is stored in the general group in the NWB file. And so for extracellular electrophysiology, this includes metadata about the electrodes that were used for recording, which are stored in a central table in the general metadata group. And so that's where then you also have additional metadata about the devices, the subjects, and so on. And so at the next level then are the recordings of the extracellular electrical activity, which are stored in electrical series as part of the acquisition group. And so the electrical series then also explicitly references the relevant metadata, which in this case are, what are the electrodes that were used for recording a particular electrical series. So in that way, the data that is being recorded is also always linked to the actual metadata that further describes that data. And so NWB then also supports storage of primary preprocessing results. So here in the form of detections of spikes and other events and features in the data, which are stored in the processing group. And then finally, in the context of extracellular electrophysiology, NWB also supports storage of results from spike sorting via its units table. And again, NWB here explicitly links the data to the relevant metadata descriptions here in the form of the electrodes and electrode groups, as well as the raw data that was used to extract those spikes. And so NWB, in a very similar fashion, supports storage also then of optical physiology pipelines, which are in the same manner organized into general imaging metadata, acquisitions of raw and motion-corrected time series recordings, primary processing via image segmentation and detection of regions of interest, and storage of derived neural activity, such as fluorescence traces from MRI. But NWB is more than just a file format. It defines an ecosystem of tools, methods, and standards for storing, sharing, and analyzing complex neurophysiology data. And so here you can see in the figure an overview of the NWB software ecosystem. So at the foundation, we have the NWB data standard schema, which defines the rules of how to organize experimental data and metadata in NWB files. It defines, which data is required, how to call data fields, and so on. And then you have extensions to the NWB data standard, which are then managed in our NWB Neurodata extension catalog, which you can find online. And then using the PyNWB and MetaNWB reference API allows users to easily create, read, and write and use NWB files while ensuring compliance with the NWB data standard. These tools also include additional tools for extending NWB, for validating NWB files, and other essential functionality for using NWB file and interacting with the data standard. The higher level tools shown in red then facilitate conversion of data, introspection, exploration of NWB files, creation of documentation, and extension, among others. And then finally, there's a growing collection of community software for data analysis, visualization, and management that use NWB technologies and provide additional rich functionality to end users. And so NWB technologies are really at the heart of the neurodata lifecycle. Data standards are a critical conduit that facilitate the flow of data throughout the data lifecycle, from acquisition of the data, processing to analysis, and publication to preservation, and then finally reuse. And they've also facilitated the integration of data and software across these different phases of the data lifecycle. Right? For example, by having support for a single data standard across the data lifecycle, we can avoid converting data between different phases of the lifecycle. We can now take data from multiple steps in our processing pipeline and integrate it into a single file and share that file with our community. Right? But what it means is that NWB needs to support the needs of and integrate with technologies across all these different phases of our data lifecycle, from acquisition to analysis to publication. And so our goal with NWB is really to work with, not compete with, existing and emerging technologies in the community. And so a large part of what we do is really working with the community to build out this ecosystem of neurophysiology tools and really provide value to the community by having NWB help integrate across these different phases. And so finally, NWB is a data standard for, not a data standard of, neurophysiology experiments. And what I mean with this is that with NWB, we do not aim to standardize neurophysiology experiments itself, but we aim to standardize how we save and share data from neuroscience experiments. And so a key part then is, what are the tools that are available to you as you endeavor into reusing NWB data and analyzing data that exists in the DANDI archive. And so to learn more about the NWB core software, as well as the community analysis visualization tools, I encourage you to visit our NWB overview.read2docs website, where you can find an index of core and community tools. And also, if you have a tool that you've developed that supports NWB that you want to share with the community, you can also let us know and add it to the index to make sure other people in the community are aware of your tool. And so to give you a quick overview of the main tools, I'll here divide the space of tools in this slide along two main dimensions. On the one hand, there's the programming environment, Python, MATLAB, web, and other, and then also the main neurophysiology applications such as extracellular, EPHIS, optical physiology, intracellular electrophysiology, and behavior. And so I'm going to start with tools that are more cross-application targets, and then go to more tools that are more application specific, such as tools for electrophysiology or optical physiology. And so the first set of tools that you will likely encounter in your endeavor with NWB are the PyNWB and MetNWB reference APIs in Python and MATLAB respectively. And so using these APIs, you can read and write, validate NWB files, and then in the additional tools such as our NWB inspector, the neuroconf conversion tools, and tools for extending NWB really help with validating and conversion of NWB data and creating extensions. And then next is the Distributed Archive for Neurophysiology Data Integration, DANDI, which is a cloud-based platform for neurophysiology data storage and sharing. And you will learn more about DANDI in Satra's talk next. And so next up then are tools such as the NWB widgets and the NWB Explorer, which are really general purpose visualization tools for exploring the contents of NWB files and learning more about the structure and an understanding of the files. DataJoint then is a free and open source framework for programming scientific databases and computational data pipelines. DataJoint is useful, for example, to manage analysis pipelines. It can also be used to manage large collections of NWB files. For extracellular electrophysiology, you will find tools such as FieldTrip, which is a MATLAB software toolbox for MEG, EEG, and IEG analysis. There's Cell Explorer, which is a GUI and a standardized pipeline for exploring and characterizing single cells acquired with extracellular electrodes. There's Brainstorm, which is a collaborative open source application dedicated to the analysis of brain recordings from MEG, EEG, FNIRS, ECOG, and depth electrodes and multi-unit electrophysiology, or ECOG-Viz, which targets quality control, annotation, and processing of ECE-Viz data. Another tool that's really interesting for extracellular electrophysiology is Spike Interface. Spike Interface defines a standardized Python API that unifies data formats and spike sorting algorithms. And so Spike Interface provides you access to a broad range of spike sorters while allowing your data to remain in NWB, both for the input and the output. Another tool that targets integration across standards is NEO, which is a Python package for working with electrophysiology data with support for reading a wide range of neurophysiology file formats with a goal to improve interoperability between Python tools. For optical physiology, you will find tools such as K-man, Sweet2P, and calcium imaging analysis, which are all sophisticated tools for calcium image analysis in Python and MATLAB respectively. And all of these tools support NWB read as well as write. We're also endeavoring into working with the data acquisition system software to support NWB. So for example, MEES is a suite-based data acquisition tool used for intracellular electrophysiology recordings written in Igor Pro, which supports acquisition in NWB. And then the OpenEFIS GUI now also supports recording of extracellular electrophysiology data in NWB via the NWB data format plugin. And then finally, for behavior data, we'll find tools such as Deep LabCub and Sleep and Pineapple that now support NWB, so you can extract your animal behavior using these tools and store the data in NWB. And so this just gives you a very rough overview of some of the analysis tools that are available for NWB and that you may find useful as you endeavor in this showcase in the next week's Neurodata Rehab to reuse the data. And so we're continuing to work with the community to grow support for NWB, and we expect more tools to add support for NWB in the near future. And if there's any particular tools that you would like to see support NWB, then please let us know. Reach out to us and tell us which tools are useful for you and that you think would help provide more value to the community. And so, in conclusion, a key goal of our efforts in NWB is to really help the community overcome this energy barrier in data standardization to advance fair neurophysiology data through the dissemination of NWB and to encourage reuse and reanalysis and sharing of neurophysiology data. And so on the one hand, to overcome this energy barrier, there are sticks that provide a framework to support and motivate users to climb this barrier of adopting standards. And so we do this through engaging with NIH, the International Brain Initiative, data archives, and many other stakeholders to help create the funding incentives and the data governance principles that help us create an environment that is conducive to standardization. A key aspect of our efforts as part of the NWB project itself then is to provide guidance and support to the users and the community to help lower the barrier of adopting NWB by creating training resources and providing in-person training opportunities, by providing support for labs and projects to integrate their data and processes with NWB, and by creating community-driven processes for integration of new neuroscience technologies with NWB. And then finally, there are also the carrots by providing immediate benefits and impacts to users by reducing costs, time, and effort, by improving quality, reliability, and accuracy of data and results, and providing access to new scientific capabilities. And so one of the key aspects there is that I show that we're working with developers to promote and support integration of NWB with neuroscience tools and really develop and maintain an accessible and sustainable open source software ecosystem for NWB. And so to learn more about NWB, there's a broad range of documentations that you will find online. So at the top, you will find the nwb.org website, which provides an overview of the NWB project, our organization, our policies. You can reach out to us there on Slack. We have a mailing list that you can sign up for, and Twitter. And so those provide you with general information about the project and provide an ability for you to reach out to us. If you want to learn more about NWB to help you adopt the standard and use it, I highly recommend that you go to our NWB overview.read2docs website, which really serves as an entry point for users and developers to learn about the NWB ecosystem. And as you have questions, you can create issues on the individual code repositories, as well as reach out to us on our help desk and on Slack. And so, finally, I want to thank the whole NWB team. And I know there's lots of people that are missing here, but I want to thank our teams at Catalyst Neuro, at Berkeley Lab, the Allen Institute, many other institutions, and in particular, also want to thank our NWB Executive Board, which help guide the development of NWB as we move forward. And so, visit us at nwb.org and reach out to the team as you have questions. And so, with that, I will hand it over to our next speaker.
Thank you, Oliver. So we have Yarek Halchenko up next, if Yarek wants to share his slides. And just an FYI, we will, as we're recording this, these slides and this recording will be available on the Allen Institute YouTube channel after today. All right, Yarek, I'll let you take it away.
Now you should be able to hear me.
We can hear you, but now we can't see your slides. So if you
That's
want to try
right.
to screen share again, that'd be great.
Yes. Here we go. You can see the slides now.
Yes.
Great. So here I am, Yaroslav Halchenko from Dartmouth College, and I'm presenting DANDI Archive, which is archive and collaboration space for a neurophysiology project. It's called DANDI for that. And this project is initially supported and still supported by through Brain Initiative, and also through all the data is stored on Amazon Cloud. So they provide us storage through a public dataset portal. And this work is not only for Dartmouth, it's led from MIT, from Satra Ghosh, and we in collaboration with Kitware and Catalyst Neuro. So before we go into greater detail about DANDI, I want to introduce a new term, DANDI set. So what is DANDI set? Is organized collection of assets with both file level and dataset level metadata generated from experiment. So to some degree, it could be considered a dataset of DANDI. And what we are trying to achieve is that such a DANDI set is a FAIR collection. I hope that you have heard about FAIR before. It's findable, accessible, interoperable, and reproducible. Jesus, I've forgotten what R stands for. So we try to make sure that DANDI set is a FAIR collection. And we distinguish working with different tiers, let's say, of collaborators or users of the archive. Some of them are principal investigators. What matters to know for those is that we support lots of data types for across the board, across species. We support uploading of electrophysiology, optophysiology, microscopy, MRI, behavior, and stimulation data. And for that, we also provide services for data storage. And with that, we want also to solve the problem of data dissemination. So we are not providing just a sinkhole where data goes, but we also strive to improve data dissemination and reuse. That's why here I am talking to you, but also data publishing. How publishing is different is that we guarantee preservation of the data and availability of it later on. And also features such as search, so people could find the data for their needs. But because I've mentioned that DANDI is also collaborative space, we provide research space where people could work on that data more conveniently in our Jupyter Hub in the cloud. And with that, we strive to improve collaboration. And here at the bottom, some examples of those data modalities, which I just mentioned. What is important to know is that requirements are pretty loose, let's put it. that data, what we require is that data could be embargoed for a period of time, but then it should be released openly and typically under Creative Commons license, and especially for BICCN projects, that's specifically CC by 4.0. And we invite principal investigators also to participate, provide feedback, and better define what is needed to make their research more efficient and what other needs in the community could be from data archive, and also submit and provide maybe analytic tools which we deploy on our research space so others could use it. Terms again for sharing the data are quite open. All the data, metadata is available through CC 0 license, which is pretty much public domain dedication style license. It's not even a license per se. And users responsible for submission and to define, to adhere to the terms of those licenses. And the only other limitation is that the largest file size in the archive cannot exceed 5 terabytes, which is a limitation of Amazon Cloud. But I think we didn't exceed that anywhere yet. And then next tier of users or with whom we work through this project is data submitters. And overall, there is 2 levels, and it's not like in data reuse, you need to submit the data, but you might as well do. Let's say if you produce some derivative data set, which is valuable on its own, and you spend a lot of time to prepare the derived data, then you better share it so others don't need to go through the same hurdle. That's why I want to go quickly through data submission process as well. How typically it's done, that on the web's interface, people register the DANDI set and in the lab first, of course, people collect the data, and again, across all those modalities, but then they standardize it. So you just heard from Oliver about Neurodata Without Borders and WB, which is file format to contain electrophysiology and other data types. You heard about that, but hopefully you've heard also about brain imaging data structure, which is more of data set level organization of files. So it's how you name your files and what additional metadata you extract and provide in sidecar JSON files. And DANDI sets, some of the DANDI sets, they contain bits data sets, but the rest is just, it could be bits inspired layout. So you would go and you see that all DANDI sets, they all look very similar. So people collect data, they convert it to NWB or other formats which is supported by bits. For that, we provide some tools to organize the data. Then they go online, they register a new data set in which they would upload it, provide basic name and description for the data. And then we provide more advanced editor for all the metadata fields, which are here to specific schema, which guides you through filling out all those information. So others could easily deduce what is that data set about and how could they use it. And then we provide client tools to upload data. So uploading the data is quite easy. Again, you go first through organization of your DANDI set if you just create it from collection of files. You validate the data to ensure that it adheres to the standards, and then you use DANDI upload command while entering the API key which you can get from the website. And that's how simple it should be to upload the data. If your data adheres to the standards, validation would go through and upload will not have any concerns. If there are any concerns with specific file that, let's say, it doesn't validate correctly, the upload by default will skip that file. And then after a data set is uploaded, it's present in a draft version. So it's the version which could be changed. You could upload new data, you could fix metadata, and eventually you would publish the data so it becomes out of draft into some other version. Okay, and then how does it look for researcher? So researcher or user or student who wants to access the data. So on the archive, this screenshot is a little bit outdated. We can already contain around 400 terabytes of data and around 200 DANDI sets. You could quickly navigate through the website by going and exploring the DANDI sets and different sorting. And to get the data, you don't have to log in. So all the data which is available and not embargoed is available for free, and you don't need to create an account to just access the data. And while exploring, you could navigate and see that metadata, which I've mentioned before, and also see individual files. And right there, you could even see the metadata for those individual files by clicking on this blue eye, or download individual files straight from the web interface by clicking this download button. You could also see the sizes for the files, and with triple dots, you could possibly open additional actions. Let's say for NWB files, you could open that file on NWB Explorer on Metacell. So you could explore data sets, you could download them already from the website. But if you could want to download more than single file or want to do it programmatically, we provide at least two ways. There is also API, which I am not mentioning here, but you could use the same DANDI command line tool, or it's also a Python library, which you could point to specific DANDI set of specific version. As I mentioned, you could have DANDI sets in draft version, or after you are done preparing and want to publish that data set, you'll get the DOI and specific version. So you could point DANDI download command to a specific version of a DANDI set, and or latest or some other version, and download it all, or could download specific files or even folders. You pretty much copy URL from web browser into DANDI download command and get your folder, let's say, full of data. But maybe you want to explore more, I would say, modern and sophisticated ways, if you especially like working with version control systems such as Git. All DANDI sets are provided from GitHub as data-led datasets. I would advise you to discover more about this by going to, what is that page? It is github.com DANDI sets, which provides you more information about data-led as well. There is a handbook which you could read, and there is also a YouTube channel which you could watch, and then there is technical forum, community chat, and all those data sets, you could see that they're available as Git repositories on GitHub. So they don't really contain all these gigabytes of data. Data is still residing in DANDI archive, but with data-led tool, you could easily get specific files and use this version control way to obtain that data and include it in your analysis. If you want to discover more about these ways, you could also look at, I'm sorry about jumping. So you could look into Yoda principles, which are referenced in the handbook, how to make your construction of your analysis and workflow more reproducible later on by including everything necessary for your analysis. Let's say containers, data sets, scripts, all of that. Okay, so this is at least 2-3 ways to download the data. One is from web interface, another one using command line client, and third one is through data-led datasets. And then you could also interact with the archive by using hub of dandiarchive.org, where you could start instances of different sizes. You could see that there is tiny, base, large, and extra, and even GPU instances if you want to utilize some GPU-based compute. And all of that is available for free. And then, once again, after you used and you created some data-led DANDI sets, you could publish your work, of course, but also you could publish your derived data back on DANDI archive. That's why publish step was also important. So overall, what is underneath is also might be useful for developers if you want to work with us and develop some other additional coolness on the archive. Brief information that everything is developed in open under DANDI archive, DANDI organization on GitHub, where you could find all the repositories for all components of DANDI archive. And you could also build your own tools which would talk to our archive through RESTful API. For that, you could just interact directly with API or use our DANDI Python library, which is part also of that DANDI command line interface. So pip install DANDI brings you that. And there is, of course, view components, which for the web, which we also welcome additions to. And everything is tested through continuous integrations. You could be assured that you don't break anything, so give it a shot. And there is more links to different aspects, different standards, and we have help desk GitHub repository where you could ask your questions as well and get answers. Thank you very much.
Thank you, Yark. And we do have one question from the audience is, could you elaborate on why you would use DataLad? Is it purely for version control?
It's not purely for version control. Some people find it much more convenient to use DataLad or Kitanex directly to get access to specific portions of DANDI sets. Let's say I want to work with these files. I want to quickly navigate and get this file or drop that load if I don't need it. So even if you don't care about version control, it might provide you easy means to explore those DANDI sets. But otherwise, it is indeed for version control, not only of those DANDI sets, but putting it into version control of your studies.
Great, thank you. One other question, is there any limitation on how many users can use the DANDI hub simultaneously? Relatedly, what is the current plan for sustaining the DANDI hub, and will it always be free?
Well, let's go in the order questions were received. Consider that there is no limitations as long as you use it for good cause. Let's put it this way. And at the moment, the cost is not prohibitive us to even expand the number of pods. Underneath, it's all orchestrated through Kubernetes, so we can scale it up dynamically as need arises. So at this moment, let's not worry about how many users. We didn't kind of reach the point where it becomes a problem. As for sustaining, what we expect later on is that if a lab needs more than what we can afford, we will, first of all, everything is open source. You could start your own hub, but also we want to make it easy for people. Oh, let's start this instance on my credit card, pretty much, right? So we want to sustain it that way. So to scale up, if you have funds and do big research, then we could do it this way. Another way, if there is a demand, we could always apply for supplemental grants to direct, covering costs of compute which is needed to the community. So there is this at least 3 ways, right? So I hope that the kind of trial version will always be free, right? So people would always be able to collaborate in minimal settings without exploding, let's say, do spike sorting on some terabytes of data, which might be too intensive. I hope that answered the question.
Great, thank you. And we have a few more coming in. So one of them is, what are your opinions on the current best practice? So not just what's technically possible, but the best practice for NWB storing, sharing of very large raw data compared to pre-processed data. And so just an example, like raw and pre-processed as separate files or massive Uber files, including behavior, high-speed video frames, linking images, etc.
Okay. Well, first of all, you've mentioned already raw and pre-processed data. Because there is no golden truth how you pre-process data, right? You might have different pre-processings. And for that, you already start feeling that maybe it shouldn't go into the same file, right? So I would recommend first to separate at the level of dataset or DANDI set. So these dataset with raw data, right? And that's where we could talk also about now, should all modalities go into NWB file? And that pretty much reflects into those video files and all that. So my and Satra's idea is more of bit style, where we although NWB allows you to stick all kinds of data into a single file, although we provided means to access those files kind of more efficiently from online, we recommend to take maybe and split it apart into files more modular. Let's say this is electrophysiology data, this is optical data, right? So have separation at the level of the file. So if somebody needs to analyze only one data type, then you could get that file individual. Of course, you need to bind them together in some logical semantic way. That's where bits or something like bits could allow you for that kind of binding at the level of different files. Then for some data types as video, it may make sense to keep them in the original video file outside. And I think we have already support for that in NWB, that you could provide external links to the files. So by storing them outside in native format, then you could use tools right away, which are not even aware of NWB, but aware how to play, let's say, AVI file, right? So I think best approach is to separate out different modalities, even for the same recording session, possibly into separate files, and use data set level organization to kind of combine them. And then for derived data, I would even say that might be even separate data set. And that's where Yoda principles kind of helpful, where you say, I am pre-processed data for this DANDI set, and that DANDI set is linked to me as source data folder or sub data set underneath. And then people could easily get to the source data if they need to, but they could also get only derived data if that is the goal. I hope that answers at least some part of logistics, but this is a whole big field, you know, what would be the best practice? If an article published on the data, how do the references, authors, and credits are affected? If an article published based on the data, how do the references affected positively? I would say. I don't know in what sense to answer this question.
And it might be asking, and please correct me if I'm wrong from the questioner, how do you deal with the references, authors, and credits if publishing on the data reuse that you're using here?
I would, well, first of all, typically in our field, we are trying to stay as open as possible and invite all co-authors who, let's say, had to do anything with the data set to be among co-authors. And if you look at the DANDI archive to get cited by, I believe you will get all the authors of the data set listed in that, let's say, DOI record. And then when people cite that DANDI set, they will get all the authors which were listed for that DANDI set cited. So I think, and then what is the positive? Again, you get two citations. If you publish your DANDI set or data set on DANDI and then prepare data which describes that data set, so it would be data descriptor paper, and most of the time, let's say Nature, scientific data requires you to deposit first data into public archive, right? So then you have two publications. You have publication of the DANDI set itself, DOI for it, and then you have publication of the paper describing that data set. So you have two publications for kind of the price of one.
All right, thank you very much. If there's no other questions, next up we have Jerome Lecoq talking about the OpenScope program. So if Jerome, you want to load up your slides. And just a reminder again, please enter any questions that you guys have as participants in the Q&A box. And then we are also recording this and it'll be on YouTube and the Allen Institute channel if you guys want to share it in the future. So Jerome, if you want to go ahead and load up your presentation.
Hold on, I have to re-enable my computer is new.
No worries.
It needs the permission to access the PowerPoint.
And then, so Jerome's going to load that up. Afterwards, we are going to have a short break at 10:30. And when we come back, we are going to have a roundtable discussion at 10:45.
I need to log out and log in. I apologize.
Sure. And bear with us as Jerome sets that up. So yeah, we'll have an overview of the OpenScope program, which helps teams apply to be able to propose their own experiments to be run at the Allen Institute and then produce data on that. That data will eventually be shared with the rest of the community. So it's a great case example of new data being produced that can be reused by others. At 10:30, we're going to have that quick break and then come back for a roundtable discussion on the pros and cons of reusing data sets, questions we should be asking along that process, probably related to that same question on publication and giving credit to others and how should we best do that, and then any caveats to this process. And then at 11:30, we're going to have this data blitz of others who have done data reuse in the past. And then finally, we'll have that poster session in Gather, and we'll be sharing you guys the link to the Gather within the Zoom webinar. All right, Jerome, are you ready to take it away?
Yeah, is my screen okay? Is it
Yes,
shared?
we can see your screen.
Wonderful. I had to, it's a new computer, so I had to re-allow Zoom to access the video stream. Good morning, everyone. So my name is Jerome. I'm an investigator in the MindScope program in the Allen Institute, and I'm going to talk to you about OpenScope today. This is a community effort. And so before I dive into the talk, I want to make sure I emphasize that there's a lot of people behind this work, not only me. So there are scientists, research associates that are collecting data, managers that are managing this process, engineers that are building the pipeline, animal care. We're talking about 100 people, individuals. I've listed some of the scientists below that contributed to building what I present today, but I'll make sure that you take this on. This is really a team effort. There's also a steering committee behind the OpenScope program that I listed the wonderful individuals that are contributing to this, and we are under NIH funding, but sort of support us in sharing this resource to the community. So the community is very central to that project. It's a key part of it, and so that's why I want to start with this. Then I'm going to talk about the OpenScope project, what it is, and then I'll mention a brief description of what the pipeline is behind the project. And then we will talk about the yearly request for proposals that we put out. And then I briefly mention what does it mean in practice, and talk about NWB files in particular. Okay. So what is OpenScope? You've seen the success of the observatory model in astronomy. Very recently, they push out a new Webb telescope that is blowing the mind of everyone, and it's been used by the community at large. Here I have a graph taken from a publication from the Keck Observatory in Hawaii, where they show the number of publications over the years coming from this observatory. And this is essentially data reuse in many cases. So in astronomy, they push data set out there, and then some of them collect new data, some of them reuse surveys that they made. And so we thought that there is an opportunity in neuroscience to push that approach. And that's part of the goal of this little workshop. So a few years ago, we seek to replicate this model and build a pipeline around the astronomy model, standardize the process, and make it eventually with the idea that we'll make it available to the community. That was a vision that Christoph Koch brought to the Institute. And 2 years ago, we sought funding from the NIH to open it up to every scientist across the community. And so that's what OpenScope is. It's a U24 resource, and we'll have a yearly application cycle. It's within the MindScope program at the Allen Institute. And what we do is we open up our 2-photon and NeuroPixel experimental platform for new experiments that are proposed by the community. Our long-term goals is to first maximize the impact of our platforms from a scientific standpoint, and then, and importantly, build and support a community around this observatory, which is also a goal of this workshop, and help develop the ecosystem. So one of these aspects is the reuse ecosystem around datasets. So every dataset that we collect as part of the OpenScope project gets packaged into NWB files, pushed to DANDI archive, and we want to build a community around this, and so the hustle to sort of show the light and help other labs do the same. So what is the data collection pipeline that we are giving access to? This is a short presentation, so I'm not gonna have the opportunity to go into too much details, but it's a hand-to-hand data collection pipeline that start from transgenic animals and finish with web product. And we enable surgical implantation of implants. So for 2-photon imaging, that will be glass windows. For a NeuroPixel recording, that will be a pre-calibrated implant with holes to position the NeuroPixel probe at precise location. We have both passive and behaviorally trained animals now. And then you can use either the NeuroPixel or multi-plane 2-photon calcium imaging. That's the step in the middle here. And then we take the brain out, reconstruct in 3D. For the NeuroPixel, we register to the Common Coordinate Framework from the Institute. and check for brain health and so on and so forth. So it's a whole process. And as you, if you get access to a project at the OpenScope and design an experiment, we'll essentially do everything except the experimental design and the stimulus that the mouse is presented with. So that's the first component, but that's not the only one. The second component of the resource is this yearly application cycle. We just finished the second round LOI, the letter of intent. Typically, we start in the summer, so the next one will be next summer. It's a double-blinded review process to avoid any potential source of bias, and it's open to the entire community worldwide. So that's the second component, to make sure that we serve the community at large. The third component is an integrated data ecosystem around in the cloud. And so for this, we don't want to reinvent the wheels. And so we'll, as much as we can, we'll leverage the tools that the community is building. And Neurodata Without Borders and DANDI archives are currently our focus because they built this ecosystem. And I talk a little bit about this in the end of the talk. And the last component is a technical hub. That's important to me. So to build this community around the observatory, we need to sort of broaden our impact and reach. So the first to achieve this, one component is this online workshop that we're having today. And we have a hackathon next week for people that were able to come to the island and propose a project. The second component of this community is a technical workshop that we had last week at the Allen Institute. So similarly, people from all across the world could apply and present their interest in learning our experimental tips and tricks, how we acquire the data. And so we presented that last week. So it was a 2 to 3 days event, also in collaboration with other U24 resources that are funded by the NIH. Okay, so what's the proposal cycle like? So as I mentioned, that we are currently in the middle of the second round. This is a yearly process. We will have 4 proposal cycles over the next 4 years. The first round last year selected passive script. So we are sort of increasingly adding new technical capability to the project. So it has to provide more flexibility in the kind of experiment we can run. From the start, we enable both 2-photon multi-plane imaging and neuropixel recording. This year, we are starting to add behavioral capabilities to the call, as well as a broadened neurosurgical reach, so recording from more brain areas with neuropixel. Next year, our plan is to expand the areas you can record from with 2-photon as well, so from the back to the front of the dorsal skull, and allowing studies of sensory motor conversion from 2-photon standpoint, as well as continue working on the behavior aspect. And so this will be a yearly application process. So what is this pipeline? I mentioned this. I'm going to briefly talk about what are the capabilities here. The first thing is our system is highly standardized. So as you can see on the top left, the 2-photon on the right and the neuropixel recording rig on the left are almost equivalent, except for the recording modality. The mouse is seeing the same standardized condition. And this is enabled by a large amount of work on CAD from the engineering teams to standardize the data collection process. We've shared a lot of our design on this web portal. You can get there and learn about the tools that we built for this. The workshop last week was also one of the goals was to facilitate this dissemination of our pipeline development. A key component besides the CAD of the standardization is sort of a shared coordinate system from the mouse implant to the recording day, which is sort of illustrated by this picture on the right. which allow us to position recording targets based on, for instance, functional intrinsic imaging, which you have an examples on the bottom right. So it's a very standardized pipeline. I mentioned that we are able to do multi-area calcium imaging. So this is an example data set where you see 8 plane of data were acquired at 10 Hertz across 2 cortical, so you could say 2 cortical column from the layer 1 to the layer 5. And so we can position this really to enable many kinds of studies. The NeuroPixel recording capabilities was developed over the years. We are currently positioning 6 probes in the visual cortex, but this year we are accepting projects that explore beyond the visual system because we iterated our surgical protocol. Again, if you're curious about this, I encourage you to look at the request for proposal document that we pushed out this year. The new component for this year is the detection of change. So our current task is a mouse is trained to it's a go no go task, and the mouse is trained to detect when a new image is presented after a number of flashes, and then it responds with a lick. For the OpenScope project, we're starting with training the mice on this task and then enabling modification of the task on the rig, just to help any sort of allow studies of error or expectation or so on and so forth. So we'll continue to work with this task in the future and expand it or modify it a little bit based on the community request and the steering committee feedback. So I mentioned pipeline. As I said, it's a lot more to talk about, but for the interest of time, I'm not going to dive too much into details. But we have a lot of a document on the pipeline. We have a couple white papers you can consult on the Allen Institute web portal, as well as the request for proposal document that is currently out on the OpenScope web portal. Okay, so this year, just to give you an examples of our process, we are seeking 3 proposals that mixed passive or behavior capabilities, as well as NeuroPixel and 2-photon. That's the second line here. And it's a leverage boost both of those capabilities, the NeuroPixels and the 2-photon imaging pipelines. And you have to select one of these, as well as variants of the detection of change. So what you get when you apply, and so you can go through this process. You sort of review by your peers. The double blinding process is done by external reviewers. And if you're selected, you will get a number of dataset from 10 mice. In 2-photon, this will be up to 90 curated datasets. So we collect more dataset than you need. And those files will be delivered to you as NWB files. The dataset will be embargoed if you desire so for a year. It will be directly on DANDI archive. We're doing this right now with the project we selected last year. And then the goal is to share this dataset for reuse by the community, which is a key goal of this event. In addition to this, we think it's important that you'll come on site to learn about how the data is collected. We just went there with one of the team 2 weeks ago. One thing we're trying to do in the future is to help develop this compute ecosystem around the NWB files. And I talked to this in a couple slides. Okay, so how does it work in practice? So these are the future calls. And in practice, the next call is going to be next summer. And so what I'm hoping is that as you develop your data reuse project, and if you leverage datasets that are present on the DANDI archive, think about if you're missing some aspects in your analysis or in your experiment, you can apply to the OpenScope project and submit experiment based on your analysis. You don't necessarily need to collect the data yourself. Maybe it's actually a better use of our pipeline than just building everything from scratch yourself. So it will depend on your project, but there is this opportunity that is up to you if you decide to so. These are the projects that we selected this year. I thought it would be helpful to just go scientifically through them so that you get a sense of what kind of questions you can answer. The first project on the top left is coming from Vanderbilt University. Was proposed by Alex Meyer, Andrew Bastos, and Jacob Westerberg. As I said, Jacob was there at the Allen 2 weeks ago to sort of see our pipeline. And they are interested in comparing neuropixel recording in mice with monkeys. So they have some recording in monkey, and they want to compare the responses there. That's the first thing. The second aspect is they're interested in the coding of oddball responses. So these are when you learn a model of the world and it's being shaken. There are two kinds of oddballs they're interested in. Is this local oddball? So when you see a sequence of image and it just changes suddenly. So you have a local model in your cortical tissue of this sequence. If you alter the orientation of the grading suddenly, it's likely going to break this model and trigger responses. Alternatively, if you train the mice to always see that sequence, and at some point you change it, it's similarly you're likely going to get an oddball response. And that will likely involve a larger set of areas because it's a broader and more complex training. And so they hypothesize that this will involve a larger hierarchical set of cortical area. And so we are testing this hypothesis in this project. The second project that we selected is about, so it's from Berkeley, from Yi-Young Shin and Lola Desnik lab. So Yi-Young has acquired 2 photon dataset in this task, and she was interested in comparing the responses with neuropixel recording because it provides very complementary capabilities. And she's using illusion as a proxy to look at the binding. So when you have two stimuli that are separate in the image, and sometimes they form concepts across like an object. And she's presenting visual stimuli with and without that illusion. Presumably, sort of when the illusion happens, it means that there's binding occurring. And she has hypotheses on how this happens across different cell types and frequencies. The last project we selected is related to a previous OpenScope project we ran multiple years ago. So there, it's coming from Northeast Canada, from Blake Richards and Joel Zilderberg. So they're interested in comparing somatic and dendritic responses, again, with like a kind of stimuli that is similar to the oddball on the top left. But they are more sort of, they want to dive into the cellular responses, and they have some hypotheses on the role of dendrites and somas into this. So we're acquiring 2 photon data set where we can relate exactly the same cells with multi-plane imaging. So, and these projects are coming from a mix of experimentalists and purely theoretical scientists. So it's open to both types. And as you see, some of them have preliminary data, some have not. Okay. There are a couple of NWB dataset that we have already released that I want to highlight here from previous years. The first one is this meaningful project where we presented many different types of natural and artificial stimuli. And they were interested in testing some metrics of consciousness when the mice is seeing images that are relevant to it and when they're not. There's a paper that I've linked here from Will Manor and his collaborators that test this, but you can access this data set. The second one I want to mention is this previous data collection on the credit assignment project where we acquire multi-plane imaging, but in separate sessions. Colleen is going to be here in the database, and so you can talk to her about this data set. One thing that I want to highlight that is really dear to me is she put the effort into sharing the code to regenerate the figure of their paper. And so with Carter, which is on the team of the OpenScope team now, we took our notebooks and moved them to DANDI Hub. So DANDI Hub is this notebook environment that you just heard about. And now this notebook we can rerun them and regenerate the figures directly from the NWB files on DANDI. This is something that the OpenScope team is gonna put resources on to develop in the coming years, where every project, I'm hoping that they can share with us notebooks that regenerate some of their analysis. That's the first thing. The second one is we're gonna also develop simple analysis of the NWB files we make available to the OpenScope community and beyond in collaboration with Ben Dishters and others. The idea is to sort of help develop this NWB ecosystem of analysis so that when you dive into a new NWB files, you don't have to redo everything yourself. You can just go to this GitHub repo with a lot of notebooks and then figure out the one that might be insightful to your experiments. And you don't have to sort of replot receptor field analysis or other things that you might want to be interested in, like high tracking. This thing will be, and that's my hope, that this thing will be already written. Okay, there are a lot more datasets that are available even beyond DANDI, but are in NWB files. I do want to mention that you can go to the web portal of the Allen Institute. We have dataset from that involve behavior, multiplane imaging, neuropixel, 2-photon imaging. Some of this were submitted by Saskia and her team, or Josh Siegel and others. They will be here in a data blitz, but I do really encourage you to go there and check it out. We have seen a ticking up in publication around a resource. This is just a closing remark. I think the field is converging to realize this vision that by making dataset available, you really sort of help build a community. And now we're touching close to 100 publication around the Allen Institute dataset. And so that's really encouraging that this sort of reuse ecosystem is coming together. And I'm hoping that now bringing even more data producing lab in the mix, it could really sort of spark. Okay, to summarize, I hope I've shared with you this OpenScope new model, and I'm excited and interested in looking more into this. We discussed details of the RFPs, and there are many more datasets to come. I mentioned some of these. Some of these datasets are currently in the embargo period, and they will go out on the DANDI archive. I thank the people at the beginning, but this involves the entire institute, and in particular, I'm thankful for the vision from Paul Allen. All right, thank you.
Thank you, Jerome. We had a couple of questions in the Q&A, and the first being, does the OpenScope project have to be about the cortex, or is it possible to obtain data sets from areas like the brainstem?
Yeah, the brainstem might be a little too deep at the moment. They are, so in the RFP this year, we are enabling new brain areas for neuropixel recording. So that contains subcortical areas. We list those, so I invite you to look into there. If there's a particular area you're interested in, you can reach out to us. I cherish those feedback because it helps us evolve and serve the community better.
One other question, is the letter of intent necessary for an OpenScope application? In other words, did we miss this year's application cycle?
Yeah, this is our process. The goal of the letter of intent is to limit the amount of work of everyone when we request for full proposals. And we could evolve in the future years. We could have sort of scheduled this earlier, these events, but we had to have this cadence of selection. Our next round is in the summer.
And I will add to that, the goal for the letter of intent is to give a chance for our internal staff to review everyone's proposals for feasibility really quick and make sure it makes sense to invite it for a full proposal and get additional details. So part of that process is just us having a chance to review the feasibility of the proposed project. All right, one more question. Do you have educational modules using specific data sets for engaging graduate students and postdocs that we can implement at home institutions? If not, do you offer awards to develop course materials?
I think there's multiple. So Caitlin, but organize this, is this something that is dear to her? And I think they have material for classes. We have notebook associated with our Allen dataset on the web portal that you can look at, that explain how to use dataset. If something is missing, perhaps Caitlin is the best person to reach out. Caitlin Casimo.
Okay, thank you,
I don't
Jerome.
think there are awards, as far as I know.
No, but I think Caitlin in the communications department is the best person to answer that as far as general educational materials we might have. Okay, well, if there's no other questions from the audience, we have a scheduled break at this time. Please feel free to continue asking questions in the Q&A box. So we are going to break until 10:45, and then we'll come back for a roundtable discussion on data reuse, and mainly be discussing the pros and cons of reusing data sets, questions to ask, caveats, etc. So that will be from 10:45 to 11:30. And then at 11:30, we are going to dive into our data blitz with 1 to 2 minute presentations from about 12 speakers. After that, we are going to hold the online poster session in Gather. I am going to enter the link to Gather in the chat here if anyone needs that before the break. And you'll just be able to click that link for at noon to go over to that different website. So again, please let us know if you guys have questions in the Q&A, and then we will see everyone at 10:45 for the roundtable. All right, so if Saskia, you want to go ahead and get started?
Yeah, sounds good. So I'm really excited to lead this panel discussion. We've gotten a couple of great scientists here to talk about some of the challenges with sharing data and reusing data, and things that that we kind of need to think about and discuss as we kind of venture into this space. So I'll just briefly give a brief introduction, and then I will turn it over to our speakers to elaborate more. But as you know, Ben in his opening remarks mentioned, there's a lot of opportunities for reusing data, and there's a lot of benefit and power that can come out of taking advantage of a lot of the open data. But along with that, there's, we have to kind of think carefully about some of the types of challenges in how we can find that data and use it in a meaningful and effective way. And so we have 3 panelists here today who are going to talk about their experiences using data as well as sharing data. These are 2 sides of the same coin. And so the challenges in using data are also challenges that have to be addressed as we share data. And so I think it's really valuable to kind of have both sides represented here. So our panelists, I'm just going to go left to right as how people are on my screen. Bing Brunton is a professor at the University of Washington, I think the Department of Biology, if I'm correct. Yeah, doing really cool stuff that she'll tell you about. Mackenzie Mathis is a professor at EPFL in Switzerland. And Colleen Gillen is a graduate student at the University of Toronto, who I hear is very close to finishing her dissertation right now. So yay for that. And was part of one of the OpenScope projects that Jerome was talking about a few minutes ago. And I think will tell us more about her experiences with that. And so I wanted to start, and we can maybe go in that order. If each speaker can just kind of give a little bit more introduction about themselves and their work and kind of what they've done in this space of data sharing and data reuse. So Bing, we'll start with you.
Thanks, Saskia. Thanks for including me in this discussion. I'm super happy to be here. So a little more introduction to myself. I am a computational neuroscientist. And I have this weird position where I kind of sit in between the Department of Biology at the University of Washington in Seattle, and I have a part-time appointment in the data science institute, essentially. And so my research is at this intersection of data-driven methods, using dynamical systems and control, as well as methods from machine learning to understand neural computations, especially underlying naturalistic behavior. And so I have a variety of folks in my lab who are doing all kinds of things. Like I have a couple of evolutionary biologists who are studying non-model organisms. We have applied mathematicians who are writing code and publishing it, but mostly for just open access. And then I have some people who are super into software engineering, and they're writing more like production quality code for hopefully for sharing with people, and also publishing data sets with the very much the explicit hope that other people might actually use them for their research and find it useful. We've also been consumers of other people very generously sharing their data. And this includes a variety of data sets, some that we just like kind of randomly found in papers and was really happy to have found that they actually deposited their data in a repository somewhere, to very high production quality data sets, such as the ones from the Allen Institute, where again, we were just really happy to have found like all of these really tremendous resources that make it so that, you know, since we are a computational lab, we don't do our own experiments. And so in addition to having collaborators who are actively collaborating with us, where we can talk to them, it's really nice to be able to find data sets that are openly available. And so through these efforts at publishing data, as well as trying to consume other people's data, we've definitely, I think we run the gamut of different headaches that you can run into in terms of all the extra work that's involved in trying to publish data set. That's like work that you didn't necessarily want to do, as well as occasionally you find a data set that is just, you know, so difficult to try to get a hold of, or to even figure out what it is that they're actually doing when you open it up, and you just sort of give up, right? So I think we all live in this era of social media and YouTube videos and just like this overwhelming choice. I think maybe some of our attention spans are kind of low right now, where if a graduate student opens up a data set and can't get it working within a day, they kind of give up, right? And so that's what we have to realistically fight against when we're on those of us who are producing data sets, is like you have to make it so that people can immediately open up your tool, open your data set, run your API, and actually get something running. And of course, Mackenzie and her team in developing Deep Lab Cut have like, I mean, they're the gold standard of that in our field, right? Like that is the tool that everyone goes to for lots of reasons, but one of the reasons is that it is so easy. They've made it so easy for somebody to just get started, even though they don't necessarily have all the deep expertise. And so I think that's the thing to which we all aspire.
I don't even know how to follow that up. That's really flattering, but also I just want to say ditto, right? Like everything you just said, I'm like, yep, exactly. Fantastic. So maybe by introduction, as Saskia said, my name is Mackenzie Mathis. I'm an assistant professor at EPFL, and I wear kind of two hats. My lab does do some wet work, and we collect experimental data, doing 2-photon calcium imaging and a lot of behavior, which I think will also be a big thing about challenges of sharing data and metadata. But we're also consumers. So we build machine learning algorithms and tools that we hope can be usable and user-friendly. But as being said, there's always corners to smooth out. And so we've been really lucky to also kind of discover, looking at publications, or the Allen, of course, I think has been the gold standard for data sharing for many, many years, from the Atlas to full on through the work like from you, Saskia, and of course others. And so, yeah, we think a lot about behavior. We think a lot about neural dynamics. We think a lot about adaptive behavior, so things that are changing constantly, both in true behavior, but also in the neural dynamic space. And so I think we can also speak to some of the challenges and opportunities of using others' data, and also trying to package usable pipelines of our data. And kind of, we're actually undergoing a bit of this process and just seeing firsthand and really having a deep appreciation for how hard that is to do really well. So yeah, really honored to be here. Thanks so much for having me.
We can't hear you. Hold on a second. Colleen, we can't hear you right now.
Can you hear now?
Yes.
Okay.
Thanks.
I got a nice microphone, and yet it never wants to make me audible. So thanks so much for inviting me, Saskia. I have been a part of this ecosystem now for most of my PhD, and it kind of revolutionized what my project ended up being. As Saskia said, I'm finishing up right now, be heading to do a postdoc at Imperial College in a few months. But basically, early on in my PhD, Blake and Joel applied for the OpenScope project, and we were part of the pilot run. And I ended up following the whole thing until where it is now. It's been very eye-opening. It's been difficult, but also very rewarding. And I think I've seen a lot of the challenges that you face on the grad student side when you're told you're going to be part of an open science project. It's a lot of work coming up for you. And I think it's really worth it. It's something that I ended up making a full chapter in my thesis, how we turned our data into publicly available data, how we made sure that our analysis code was reusable, how we made sure our stimuli were reusable. And as Bing pointed out, there's a big challenge there of making sure that somebody else can jump into it and actually find it worthwhile. I think I've learned a lot of things along the road. There's a lot of things I would do differently looking back. I'm really looking forward to hearing different people's perspective and sharing mine. And I really hope that this is a path that other grad students will be able to take, hopefully with fewer and fewer obstacles in the way, and that the future will be that not everyone's collecting their own data, but we're actually reusing the precious data that's being collected by the community. So thanks, guys.
Great. So I think one place where I want to start is I'm curious to hear kind of how when you're looking for data to use, how it is that you are able to find and identify that data. To what extent is it driven by here's a data set, what can I do with it, versus I have a particular question, let me find data that exists in the world that will let me speak to it. And so I'm curious what types of experiences and what types of tools and approaches you've used to find the data that you want to work with.
For us, it's been much more the latter of your options, Saskia. Usually it's a project that we already have going on and a set of questions that we're already asking. We do a combination of just searching on the internet, looking for published papers on the subject, asking all of our friends, but hey, do you know anything? Like, you know anyone who has this data set that we might be able to use? And that's how we found it. It's maybe not the most efficient way of doing it, but I'm not sure there's a better option right now.
Yeah, I think I can mimic that experience and maybe add one other perspective, which is that so in our group, we do a fair amount of computer vision as well, which I didn't mention, ironically, given that's part of Deep Lab Kit. But for that, it becomes quite interesting because the culture in that field is actually people gather around common data sets. So like huge data sets like ImageNet and these types of things. It is really about open data that then people build algorithms on. So it can be from that perspective, easier in some ways than like hunting on the webs for random data sets that you think might be perfect for the problem. But also, of course, just looking at these very popular data sets have major limitations, right? We know, and we've probably all thought or heard a lot about biased data and avoiding these pitfalls, or at least try to mitigate them as best we can. Some of that's a bit unavoidable. In recent work, when we've thought about like joint modeling of behavioral data and neural data, that actually we try to think very broadly about the field of neuroscience and kind of step back and take this meta view about like what systems and opportunities might be there where we can sort of broaden our scope of thinking. And then if you been to SFN or FENS or any of these major conferences, it's sort of hard to not miss this whole section on vision and this whole section on the hippocampus. And so then it kind of made sense for us to really like hone in and think about some data sets that had been used or published and we know something about it. So I think that becomes a key, is that it's very hard to jump into like completely unknown data sets. And so maybe that, again, I keep bringing this notion of metadata, or like understanding what the data has been used for, how it's collected, all of these things. On the other side, away from neurons, with thinking about like animal behavior, things like this, it also gets very complicated because people don't always write down how the data is collected and all the nuisance variables that you might want to account for. And so in the computer vision space, where I have like a very clean job of saying, I want a key point. That's a nose. I can identify a nose. Like this is much easier than trying to understand like truly animal behavior, right? Because there's so many variables that become unwritten. So I think that's a whole challenge of thinking about how we share data and what we ultimately share, right? Everything from like raw pixels to like the metadata is a huge endeavor. And so Colleen can probably speak to a bit about this from your experience too.
Yeah, I completely agree. The metadata point is key. I was searching through CNS data sets a while back. You know, it's a wonderful resource. I'm so pleased that they put that together, but it's not easy to search through it. It's not, you know, it's very text form. It's not particularly search friendly. And in the end, you always have to go read the paper to know as many details as you can, but you want a filter system. And that's one thing that stood out to me with DANDI when we started using it was, you know, there's some constraints on the data publishing side that are necessary. You have to force the publishers to do some of the legwork to save a lot of trouble to everyone who's looking for a data set that they can work with. So yeah, I definitely agree. I haven't ended up using much other than direct collaborations, and that's always a challenge. I think the biggest challenge I had using another data set was when I received my OpenScope data set, which at the time was in the Allen Institute data formats. That was hundreds of files per sessions. And that was more than a day of figuring out what was what. It was worth it in the end. And now I know that the OpenScope project is delivering NWB files, which is fantastic, but it's difficult for sure.
Yeah, no, and I think a couple of you brought up the importance of metadata, which you know, we were talking earlier this morning about the FAIR principles, think that data needs to be findable, accessible, interoperable, reusable. And one of the things that always strikes me if you read kind of the documentation on the FAIR principles is it's all on a huge foundation of metadata. Like all of those things rely on having rich metadata. And one of the challenges that I feel like I've faced is what actually is rich metadata. And I think we often think like, oh, I've got my species and the sex of my animals and their age and great, like done. And like that really doesn't tell you very much. And thinking about what are the ways that we can kind of emphasize and bring that metadata to the forefront, that you learn more about the procedures and the history, because I think a lot of, and kind of what I'm hearing from all of you a little bit is that you still have to spend a lot of time going back to the papers. Papers don't necessarily have great, complete or thorough method sections, especially when it comes to like data processing. And so there's still a lot of gaps that get dropped in understanding kind of a lot of the important details. And I think one challenge in this space that I think we're all thinking about a little bit is how to improve that metadata ecosystem that surrounds the data to really help bolster finding and really knowing what data is when you're kind of diving into it in that space.
I mean, I'm just gonna add on to what Saskia just said and say that I feel like I have one of the things that I've learned in working with NWB is really thinking about data as a living thing that needs to be maintained rather than like done, it's on the internet now, right? It takes constant upkeep. It's like a plant, right? It's less like a puppy and more like a plant. It's not like constantly wanting your attention every single moment of every day, but it's more like you can't just ignore it, right? Because it's going to die. And so I think like just in terms of the support, because the software changes, the data also needs to change. Metadata that you thought was complete, maybe like a year later, you realize, oh, like actually, I just didn't anticipate this particular use case. Somebody's asking me for this thing that I do have, but I never actually included. And so it takes constant maintenance to keep it alive and keep it useful for ourselves and for the community. And that's something that I've learned in working with these data sets and trying to keep these data sets alive, because and I think it's work, right? And I think we need to acknowledge that this is active, ongoing work. It is archival, but archival is not good enough.
Mm-hmm.
Yeah, I think that's a great point. I haven't really thought about it as the plant. I think I'm not really great at plants or puppies, so but I really like it. It was a great analogy. Yeah, but and one kind of thing that maybe is nice to bring up and think about is that the way the data is collected can be so variable, and standardization there is really critical for making this metadata accessible, right? Like, I mean, just as a very I know a pseudo common example probably now is that, we know that the sex of the mouse matters, and also the sex of the experimentalist can even have a huge influence on behavior. And I don't think I've ever seen a paper, unless it was on this topic, talking about the methods being like, and the surgeon was identified as female, and the, like the mouse surgeon or the person collecting the data was this, or these mice were never picked up by the tail because they were handled in a facility that only used transfer tubes, or this is the exact type of bedding that was used, or this cage size can be slightly different, right? Like, these things, how do you keep track of them? And maybe, as you said, like as the garden grows about the things that we might care about in the future, how do we like reinitialize that into the data pipeline? I think, the saving grace in some sense is that many of the questions, we might just be such a young budding field in neuroscience that some of these are kind of like nuisance variables that sort of get washed out in the noise of data. And that might be okay for a while, but I think, this is changing. So thinking about really incredible data pipelines, like how we're collecting this data, and it might even allow us to collect less data, like the OpenScope program. Because, if we really have these incredible data pipelines, like, in my fantasy world, I would just become like Bing, right? I would just be I'd be a computationalist and not like, you're amazing, don't take it the wrong way. But like, I wouldn't actually need to collect data in my own lab, right? Like for the questions, I think that this could really be something we could get away from. So this is a kind of an interesting thought experiment for me at this young professor stage, but enough about me. So someone else jump in.
I guess what I would add is I'm the type of person who stays up awake at night worrying about somebody using my data and not knowing that there was a design element, and then they think they found something, but no, that was just a problem with how we designed the experiment. And it just makes me realize that as much as you can get a lot out of the standardized numerics and categorization of your data, in the end, you also want to help people with that second step, which is doing the first pass exploration. And that's where I think the notebooks are so important. You want people to feel motivated to go and take a look at your stimulus design, take a look at what the data looks at, like from a high level. What were the trials? What were the trial patterns? When you visualize the data, do you notice some things that can alert you to a tiny methods component? I'm thinking of, we design an experiment that has these unexpected events that occur, and you might miss the fact that we clustered the unexpected events. You had several occurring one after the other. There's only so much we can tell you verbally, but you want to encourage people to look at the data before they jump in and start running analyses blindly. And that's where I think that extra layer, I sound like I'm doing an ad for DANDI archives, but having notebooks that you can run directly, I think is so important. People might not make the effort, especially if they're new to this kind of work, to go and explore your data. If you can have a notebook that they can run right out of the box, I think you're really improving the chance of people using your data productively and not 4 months later saying, wait a second, I went down a rabbit hole and it didn't make any sense with this data set. And then the other element is, as much as possible, if we can encourage people to contact the data producers and have that back and forth. I mean, it's a lot of time investment, but if it can pay off in some ways, I know we'll talk about incentives. I think that's also very important, because in the end, I just don't think we're going to be able to optimize data sharing to the point where no contact is needed and everything works out of the box immediately and you know everything you need to know. There's always going to be adjustments to be made.
Yeah, that's very true. And I mean, maybe just to piggyback and riff on that a bit, Colleen, I think contacting the data generators is a really good thing, but I would really urge people to do this in public, right? Like if you have, it's kind of like the old school thing, you're in 3rd grade, you have a question, there's probably someone else who has the same question, right? And so I think this is one thing that we can really take from the software community that's done really well, are things like GitHub and forums and discussion platforms. Like these should really be around shared data as well. Like this should be equally accessible and open and public. because I mean, Bing is very generous in saying, you know, but we did work really hard to make Deep Lab Cut very usable, but that still means that we have GitHub issues and forums. And so people email me and I like try to politely answer and be like, can you post it on the forum or did you search here, right? Like I try not to like, otherwise it's knowledge that, you know, the 10 minutes that I spend answering it or whatever it is not that much, but it's not then scalable. And so I think that's a really good point that you raised, like making things like easily accessible, pointing them in the right direction on how to use it. Although I'd say not just notebooks, but like reproducible computational environments like Docker containers. Docker doesn't pay me either, but like, you know, like, but like really reproducible things, right? So I think this is like, even at data collection time, it makes a lot of sense. Like when we record on our rigs, like we save the Git hash for exactly the commit that was used to generate that data on that day. And so anytime you could go back and reproduce it at that point. But I don't think this is a universal standard by any means. And I'm not saying I'm better or worse than anyone, but I think it's a different mode of thinking. And so I think people haven't even thought about some of these big challenges. And so I really loved your point of like interaction, this human component is still like very, very much needed in this world.
So I think one interesting analogy that I sometimes think about is occasionally I'll talk to computationally intensive scientists in other fields where they have very much accomplished this type of data standardization. Mackenzie's already talked about the computer vision community that have really coalesced around these common data sets and data challenges, right? Another community that is maybe closer to the basic sciences is astronomy. Where, you know, it used to be that if you wanted to be a good astronomer, you had to also be a really good glass grinder because you had to make your own lenses for the telescopes, right? Now you basically have to be a really good programmer because there are these now large telescopes that are global efforts that get sent up to the sky and data beams down and everyone analyzes it, right? And I know neuroscience has also adopted somewhat of this mindset by these brain observatories. We use the same terminology they do to have these terminologies. But I think I agree with both Colleen and Mackenzie is that like our field is not quite mature enough to really even know what we are looking for. Right, we're not ready to have an observatory in the same way that there's a James Webb Space Telescope out in L2 right now, because we don't agree what are the photons, right? Like everyone in astronomy agrees we should be out there collecting photons, right? And we can build instruments collect photons of this kind, of this other kind, and we're all like, okay, this is good. This is what we should be looking for, right? And that's not at all obvious in neuroscience. And so as much as we want to, I think there is a bit of a trade-off, right? Just because making that data truly reproducible is so much extra work, and that we have to weigh it against, okay, is this particular piece of data like the equivalent of something beaming down from a telescope of supernova that's never going to happen again in billions of years, we really need to keep it? Or is it like, okay, I'm just going to do this again tomorrow and probably do it better, right? Because it's not cost-free to do all of this extra work that we would have to do to make everything truly reproducible and reusable.
Yeah, I think that's an interesting point, and I think it kind of speaks to, you know, there are some large data collection initiatives, and so like a lot of what we do at the Allen Institute kind of falls into that bucket, and sharing data like that are kind of these large surveys in a sense is a little bit different from, you know, smaller labs that collect smaller data sets that you know, may just be a handful of experiments and a handful of mice in a very specific task and recording from a very unique part of the brain. Every part of the brain is unique, but you know, there are different scales to these data sets, and I think this is something where I think like DANDI archive is really valuable, because you can have all of that in that same environment, and where there might be some questions where I want to be able to look at a very specific task, but also look at a big broad survey that covers a wider range of areas and tasks and behaviors and things like that, and being able to kind of have both of those simultaneously, because I agree with Bing that like we don't know what the photons of neuroscience are, and we, you know, like I think we, you know, there's some questions, there's some ideas.
Sure, it has something to do with spikes.
Well,
maybe,
sure. Maybe,
probably
membrane potentials. But what exactly is that same modality, that same kind of survey that you get from the astronomical observatories, it isn't quite, we're not quite in that same space. At the same time, having a lot of different data can help us figure out that space better. And so I do see that they're, sharing the data that we are collecting in this time and using those data, I think can kind of help us better frame where as a field we need to be focusing in that way. One question that kind of popped into my head just a second ago, that might kind of be related to this in some ways is how, when data gets collected for a specific experiment, it's usually, we use this stimulus, we had the animal doing this particular behavior, we recorded from this particular brain region in order to answer question X. And I'm curious, for those of you who have reused data, to what extent has your reuse of that data kind of lined up with the initial design or intention of the data collection, and whether there's situations where you've been able to use data in ways that are somewhat orthogonal or complementary to the original design? Yeah, if that makes sense as a question.
Yeah, maybe I can start. So I think for one data set that we used was the calcium data set led by you, Saskia, and the neuropixels kind of equivalent, if you will. And so there, of course, it's different mice, and there was actually there's some differences in how the data was collected. And certainly I did not use it in the way that it was meant to be collected. So for us, very briefly, it was to develop a dimensionality reduction tool where we can compare across like calcium and neuropixels, but unless I've missed something major in the papers, this wasn't the original intention. And so while people had analyzed the data extremely well, one thing we wanted to be able to do is look at these representational spaces across this way, and then use some of the very paired stimuluses like this natural movie one that I've watched a lot of times. And so this was kind of one shared stimulus set across the data. But this I don't think was by design. But here I think what's important to say is that we also didn't try to make like an entirely de novo hypothesis to try to find something in the data what it was not designed for, right? And I think this kind of gets to Colleen's part, is that you have to be a little bit careful about exactly what was the intent of the data, and to make sure you're not just trying to find some random nuisance variable that they did not think was important to control for because it wasn't in the experimental design. So yeah, again, metadata. I don't know, I guess that's the hashtag of the session, maybe. This. Yeah.
So we brought up a moment ago kind of the importance of incentives and how we can incentivize both data sharing and data reuse. And I mean, I think there's a couple of pieces to this, right? One is the challenges of finding funding for both of those aspects, and what mechanisms may exist or maybe need to start existing to better support that. And another question in this space comes around to how we share credit, right? And this question came up in one of the earlier talks this morning of how do we give credit to the data collectors when we're reusing the data? Is it in the form of a citation? What does that look like? Is that the right way to do it? So I'm curious if you guys have any thoughts on, I guess those are two different questions, but pick whichever one resonates most, and we can go from there.
I mean, I think citations are a good start. That is, for better or worse, the common currency in academic circles. So if one is intent on pursuing more along this trajectory, then that's what matters: publications and citations. And so having mechanisms to actually publish data sets, having them be citable, publish data descriptors, and having those be citable, I think this is a wonderful development in our community that I'm seeing increasingly more often, and that's great. I also sort of encourage my students and postdocs to just very explicitly add a section to their CV, like data sets, like the other types of papers and conferences and all of this stuff that are more traditionally accepted. It's like, just because not everyone has a data sets section doesn't mean you can't have one, right? Like if you see that as one of your major academic contributions, just put it on your CV and say like, I did this. So
yeah, that's a fantastic point, because I didn't even ever think about this until, to be honest, I saw a senior colleague's CV that had this, and it was like open source code, open data. I was like, yeah, why don't I do that? Like, you know what I mean? Like, because you sometimes just don't know, like, what are the norms? And I think that's really important. And definitely the citations. I'd also say that, you know, if you're going like much beyond and getting a lot of help from the authors, it's definitely like minimally an acknowledgement, but also, you know, could spark a really interesting collaboration going forward. Like, you know, as a computationalist, as you know, this becomes a really important part of like understanding data, is like being very close to these people. And so I think that's great. And then I actually forget your first question, Saskia. So maybe I felt like it was great, and then I just honed in on the second one. So what was it?
I was asking about kind of funding mechanisms.
Yeah. Okay. Yeah, that's right. So my initial thought was I have no idea. I've never applied or gotten funding to reuse data. So I'm really curious about this, if anyone else has experience with this type of thing. And sharing data is actually expensive if you're not able to host it on like Zenodo or Fiction, like, right, if you're hosting it yourself and want to maintain a Bitbucket where people download. Yeah, that's actually quite an expensive endeavor too. So.
Well, and there's the hosting it, which repositories can help, you know, alleviate some of that. But as we've already discussed, data is a plant that requires continued maintenance. And so that's its own cost, right, of getting the data ready, which is a lot of work. And then there's also actively maintaining it, because I think if that active maintenance isn't something that we're thinking of, you know, a lot of these data sets can kind of become just, I don't really want to say it this way, but, you know, like litter on the side of the road, right? Where, yeah, we collected this, we did this, it's here, but like that's kind of it, right? And now the tools to access it have changed, and we aren't going to update it, you know. And so I think that cost, I think, is often gets ignored, even when there's mechanisms to help with the initial setup to some extent. Colleen, do you have thoughts on how, you know, funding from your perspective, especially with the OpenScope project and sharing that? We can't hear you again.
Can you hear me now?
Yeah, perfect.
Okay. So yeah, I was trying to think about that because I'm not really so much on the funding side of things. I do still rely on my supervisors to write all the grants, but at the same time, I suppose maybe a related concept for me has been hackathons, have been opportunities where you can tap into a greater set of resources and come together. And there's an incentive on the grad student side of things because you are building your CV. You can add that also to your CV. Because for us, that's a big question, you know, how much time am I investing in these projects? And is it going to get me somewhere? In my experience is that participating in these hackathon events is actually great for building your CV. It's great for those who won't be necessarily staying in academia. Of course, it gives you a path into industry. And I'm thinking that those concerted efforts are some of the best opportunities to check up on those old data sets, make sure they're still running, do your blitz runs of all the sort of minor fixes you might have to do if your environment's broken, that kind of thing. Otherwise, you know, I'm wondering if in 5 years I'm going to have time to go and take a look at my old data set and be like, okay, does it still run? Does everything still run? At the same time, if we're using tools like GitHub and people are actually posting issues, that is an incentive to keep things up to date. So yeah, I guess those are some perspectives I have. I don't know about how well, for example, the Canadian government is funding these kind of initiatives. I definitely see it on the side of NIH, but probably something that we should be talking about to our local sort of funding agencies and pushing them in that direction. I'm sure they'd love to find a reason to divert money away from experimental science if they can move it towards AI and the kind of resources that can be reused also on that end. Because of course, Mackenzie was talking about developing data sets for computer vision. well, that's one thing that we can start to tap into. Our data sets can be reused for that as well. I was going to mention that earlier. If you're sharing a data set that includes a full imaging stack, that's the kind of stack that can be tremendously useful for a tool like what Jérôme built, right? His deep interpolation tool. So I definitely think we just need to make funding agencies aware of all the potential that just investing a little bit more in keeping data alive can have to advance our field. That was a bit circuitous, but that's my answer.
No, I mean, I think that's an excellent point. And I think a lot of times the proof is in the pudding, right? And I think being able to demonstrate the impact that we're able to have using open data can help facilitate, the making that point to funding agencies. And I think also, I think this is where a lot of the infrastructure piece becomes really important, right? Because rather than funding kind of individuals who are using data from other individuals, if the funding is going towards the standards, and we were talking about NWB files this morning, and a lot of the software infrastructure that exists around that, both for getting data into it, but also for accessing and using it. And so putting funding into the infrastructure that helps centralize and standardize data, I think can be like extra effective at creating opportunities for reuse that then become cheaper to support, because a lot of the heavy work has already been done, and not just done once, but done in a sustainable way for kind of a larger part of the community. And so I think being able to demonstrate that impact and the effectiveness of those tools can be a really important way to kind of motivate further funding and further incentivizing of data, ongoing data reuse and data sharing.
Maybe just one thing to add here is that we also don't want to reinvent wheels just because we're a different field, right? Like there is a lot, like NWB is fantastic, and it's built on a very big data standard called HDF5 files. And so H5s, right? That is what NWB is. And so that's been used in a lot of fields, including, mini computation on other data sciences. And I think we also have to look to like, even microarrays and RNA-seq data, speaking about reuse of data. And, maybe some can fall to be litter on the side of the road, and some cannot be. But it's sort of like I was asked a few weeks ago for data that is published in a paper that's 10 years old. And I was like, yeah, I definitely bank that. Let me find that link, right? But it was just sort of a moment of like, oh my God, someone wants that data. This is so exciting for me. But at the same time, like there are these geobanks, right? So it was RNA sequencing data. This was easy to then, of course, to think about this. So I agree. We should take a lot of inspiration from fields in science that have done this well, and think about like what resources make sense for us to build, or what resources really we can reuse, I think is another just thing to bring up and maybe for discussion. But.
All right, we only have a few more minutes left. I guess the last question, maybe just to wrap things up, I'm curious if people could share what was maybe like the key challenge or hurdle that you've faced the key, I'm sure there's been several, but maybe if you have kind of a nice example of one of the kind of pitfalls that you stumbled across when you were starting to use data, or when you were trying to get your data ready for sharing, that that first caveat you would give to somebody who's starting off using a new data set. And what piece of advice for kind of getting over that hurdle would you have?
Actually, I can go first because it touches exactly on what Mackenzie just said, and that is the reinventing the wheel problem. I think it is worth it to look at what already exists. I realize now, looking back at what we put together, that we really did reinvent the wheel, and the fact is other people had made better wheels. So, it's a mistake, but if you can, as much as possible, reuse tools that are maintained, that were built to analyze data, it's really worth it, because somebody jumping in to use your code probably doesn't want to have to relearn an entirely new infrastructure that's specifically and only used in your one project. So I think that would be one of the biggest, I'd say, errors that I made that I hope to not make going forward.
Colleen, I would argue that if you learned something from the experience, it wasn't a mistake.
That's true. That's true. It's not going to be ideal for people reusing our work, but I definitely learned a lot. So
Yeah,
if you want to make that mistake, go ahead.
It's how we all learn.
It's true.
Yeah, absolutely. I mean, anecdotally, before I started my lab, I was really obsessed with being like, okay, I need like the ultimate database. I want to be able to query data in 10 years and do a meta-analysis. And I started like hacking up something, and then like a month into it, I discovered Data Joint. I was like, oh my God, I am done with whatever I'm doing right now, and I am moving on. Right? Like, and thank God it was only a month, and I still learned a lot. So it didn't feel like a failure, but
then you appreciate it, right? Because you knew how much work it would have been if you didn't have it.
Exactly. Fantastic. Right. And now absolutely. And now we have pipelines, and it's great. I'd say, like, just kind of going back to the core of your question, Saskia, for me, both on the consumer and the developer side, is like, what is the minimal viable product to get someone to engage with your data? And oftentimes it's data formatting, data loading, and like these initial steps. Like anytime you want to use a new code or a new tool, like on your data, the first challenge is like, oh, what format should it be in? How do I deal with this? Right. And so making this as easy as possible, I think, is from both parties. Or if you use a data set and you find that there's something about it that could be helpful, like contacting those authors, or in the public domain or whatever it might be, to kind of give tips, or even like repackaging it in nice ways so it can be loaded into your code very easily, or maybe a couple of ways to think about both sides of this coin. So
I'm going to add one comment, because I think it's complementary to, but a bit orthogonal to what Mackenzie and Colleen both just said. I think sometimes I struggle with especially publishing data sets, because it's like an extra effort on top of what you've already done doing the science and writing the paper, right? And sometimes it feels like too much effort, because it's not the type of work that typically, let's say, is in the purview of a graduate student working towards their thesis, or a postdoc going towards, whatever it is, the next adventure that they're going for. And so rather than not doing it or just doing it anyway, I'm always asking people, okay, so this is a team effort, because there's now more work. Like, it's not just the same amount of work, but do it better. It's actually just literally more work for you to do the science, collect data, and actually create the data set so that it is useful for somebody else. So think of it as a team effort. Like, are there parts of this that you can possibly, let's say, get an undergraduate who would love this research experience to help you do, right? Can you get somebody else to write this piece of code with you? Or is there a funding source that might fund, a small pilot effort to work with we worked with Ben Dichter for publishing some NWB data sets, because we had some funding that we were able to use them as a consultant, right? To help do this work. Like, you don't have to do this work yourself. You feel like it should be done. Are there other ways of making it so that it's more of a team effort?
I think that's a great point. I think one of the things I've learned is that open science, be it data reusing, data sharing, code sharing, all aspects of it, open science is by necessity team science. And being able to identify and leverage other people that can help you in that space, I think, is a really important, an important way to think about it. So, well, I think we are at time. Carly, I wasn't able to see if there were any questions in the chat or anything like that. I don't know if there are any. I'm
going to encourage people to keep adding their questions to the Q&A and move along to the data blitz, since that's so tightly timed. Right. So if Ben, if you want to tee that off and line up the data blitz folks. So if anyone is presenting, we have like 12 presenters for the data blitz, 1 to 2 minutes each. Ben, do you want to take that away and tee that up?
Yeah, sure. Thank you, roundtable participants. That was really great. That was awesome. And Saskia, thanks for moderating. You did a good job too. All right, so I'm gonna do screen share. Oh no, I need to share my screen and then do this. Okay, All right, it's time for the data blitz. So we have 12 different contributors that are each going to give their 1 or 2 minute presentation. I've got a timer here and I'm going to stop you at 3 minutes because we have a lot of groups to go through and only half an hour. It's nothing personal. We just need to move forward. If I stop you, I'm sorry. Just keep in mind that this is just a teaser, right? This is an uncomfortably small amount of time to talk about your work. We know that. The whole point is to get people excited about it so that you can talk over the hour and a half that you have in Gather. Okay, so without further ado, I will kick things off with Peter Peterson, and take it away. Just say next as you want me to progress through the slides. Peter, are you here?
Yes, I'm here, but I can actually present instead. Or is this fixed?
I'm going to be controlling
next. Sorry, back. So my name is Peter. I'm presenting today the DANDI set 59 with the title Theta within perturbation by focal cooling of the medial septal. This is based on a published study from 2020. Next. So the scientific question we're asking here is how hippocampal theta oscillation are coordinating neuronal activity to support spatial and memory. Next. For this, we're diving into how the medial septal is generating theta oscillation, specifically asking if by 2 mechanisms. One is of a unitary pacemaker mechanism that's imposing a single signal on the hippocampus. The other hypothesis is that it's a phase organization that's being projected hippocampus and setting up this phase relationship between regions. Next. And we're doing this by performing a thermal perturbation of the medial septal to test these models. Next. Next, sorry, expecting I couldn't control the slides. So we're doing this using a thermal perturbation device. We're implanting into medial septal. I can give you more details. At the same time, we're recording the local temperature. And then we're implanting silicon probes into the CA1 in both hemispheres. And by this, we're getting single units up to about 150 cells and LFP. Next. So the behavior consists of recording from free moving animals. Next. That's doing a spatial alternation task where the rat is running along a path called the theta maze, at the same time recording sleep. And the data set also has behavior from linear track and wheel running. Next. So the main findings can be summarized in 4 points. First of all, by cooling the medial septal, we could successfully slow down the hippocampal theta oscillations. Next. At the same time, we actually saw that the spatial representation inside the hippocampus actually remained intact. So there's no direct effect by cooling this projecting region. Next. But at the same time, we did see a behavioral effect on the animal. Next. And then finally, what we actually saw is that in the hippocampus, the compression, so meaning the time scale at which the cell assemblies are played out actually was altered by this manipulation. So these were the 4 findings. So how can you use the data? First of all, it has a number of unique features which I think are nice about this data set. It has thermal perturbation. We have temperature reading, both as a control measure, but you can use it as part of the manipulation. We have single units and LFP from CA1, and it's all in behaving rodents that are active and sleeping, and then it has this spatial memory task. Yeah, and then as extra, I can say that we actually ourselves published an extra study on this because we discovered this highly dynamic temperature. Dynamic happens during sleep as well, where we specifically looked at how sharp-wave ripples are affected by these brain temperature variations. Thank you, Ben.
All right, great, thank you. Robin, you're up.
Yeah, do you hear me well?
Yep.
Okay, so yeah, I'm Robin. I'm presenting DANDI set 219. So the main question was to describe the activity in the CR1 region of the hippocampus in developing mice, focusing on the 2 first parietal week of life. So that was like the goal, like look at how the activity evolved during development and what are the key developmental points during this early life. The experimental design is like this one. We are recording the, can you play the movie, please? On the right, I don't know. If it's not possible, it's fine. That should be like, okay, that was an example of a recording of the in vivo activity. So it speed up 10 times, but that's the kind of calcium imaging data that we acquire. And simultaneously with the imaging, we are using 2 cameras on each side of the mouse pup to record the motor behavior. The main findings are that we observed a fast transition in the integration of the sensory motor signal coming from the movement, from the limb movement to the hippocampus that first trigger activation in the CR1 region of the hippocampus before this postacetaldin-9, and that then trigger inhibition in the same region of the hippocampus. So this is acute experiment. I didn't mention that, but it's not the same mouse that we follow from 5 days old to 12. So that's the main findings. We have a paper published, and how to reuse the data. I think it can be like about like looking at how the activity correlate with the behavior. That would be like looking at things that we haven't looked at. And I also invite you to look at this Python toolbox that we developed to. So this is a NWB completed Python toolbox to do data visualization and analysis using as an input NWB files. And yeah, I'm happy to talk more about all that later with you. Thanks, Ben.
All right, great. Thank you, Robin. All right, next up is Anna Gillespie.
All right, hi everyone. I'm Anna, and I'm presenting this data set that I screenshotted here for you. This data set is tetrode recordings from dorsal CA1 of Long Evans rats performing a spatial memory-dependent navigation task. And we collected this data to perform spatial decoding of position to see how the hippocampal ensembles were replaying different aspects of the animal's behavior. So the raw data is available in 30 kilohertz sampling rate in NWB format. We do have more processed versions of this data in MATLAB, and we're in the process of moving to a data joint and Python system now. So future data sets, which will hopefully be coming, will be even more accessible. I should note that there's no single units or sorted single units in this data set at this time, but of course you'd be welcome to hook up the file to Spike interface and Spike sort with whatever sorter you wanted. The behavior data includes overhead tracking and position tracking of an animal moving around this complicated looking maze that you can see here. And we also have behavioral landmarks saved, such as lights, port triggers, reward delivery. And no matter how good my metadata is, it's really hard to get an intuition for behavior. So Ben, if you could go to the next slide and play the video, please. What you're looking at here is the overhead tracking of the animal that has a red and green diode mounted on its head for the position tracking. You can see that there are ports that light up. The animal visits ports. Sometimes it gets rewarded, sometimes it doesn't. Much more detail about the behavior in the paper, but lots of position info to be decoded here. But kind of as one of the presenters said earlier, it's hard to get a feel for this. So I'm more than happy to be contacted to talk about this behavior data and the neural data that goes along with it, of course. Next, please. And next slide. Thanks. I just want to put a plug out that my lab opens at the University of Washington in January, and I'll be looking for folks to both analyze these types of data sets as well as collect more. So if that's interesting to you, please let me know. All right, thank you.
All right, great. Thank you, Anna. All right, next up, we actually have a video that was submitted for this one. So let me just go ahead and play that. Oh, wait, I need to share this in a way that you can hear the video. Give me 1 second. Okay, that should do it.
We wanted to know what kind of input hippocampal cells born on a particular day during development receive from the medial entorhinal cortex. To answer this question, we performed in vivo electrophysiology recordings of individual cells in the medial entorhinal cortex of mice. These cells were first recorded in an open field environment where the mice were free to explore to determine their functional properties. Then the mice were moved to a separate holding box and 1 millisecond laser pulses were delivered to the cells through an implanted optic fiber to see if they expressed channelrhodopsin 2. If they did, then they would respond to the laser stimuli. Because of how channelrhodopsin 2 is introduced, we know that such responding cells project directly to cells in the hippocampus that are born on a particular day during development. This dataset was part of a larger study that you can read about in this paper. The electrophysiology data specifically relates to figure 5 and supplementary figures 3 and 4 in this paper. Want to hear more? Then come talk to Simon Ball and me at our booth.
Okay. All right, Ji Zhang.
Hi, everyone. This is Jay. You can call me JZ. So thanks for having me here today. I'm going to share this data set number 207. So next. So basically, this key question we are trying to answer here is how do neurons respond to cognitive boundaries in humans? It's kind of inspired by the previous work in rodents about the border cells, but now also the context of event segmentation. So we want to see how the brain constantly segmenting continuous experience into discrete events at the cognitive boundaries, and also how that affects the memory. Next. Then the behavior task we have is memory task. So the subjects will watch a series of clips embedded with different type of boundaries, so no boundary, soft boundary, and hard boundary. And later on, we evaluate subjects' memories in terms of their recognition memory and temporal order memory. And for this data set, we recorded in patients. So they are drug-resistant epilepsy patients, in total 19 of them. And we have approximately 550 neurons recorded from hippocampus, amygdala, and pure hippocampal gyrus. Next. So the main finding of this study so far is we found two types of cells that responds to different levels of cognitive boundaries, either responds to both soft boundary and hard boundary, or responding to hard boundary only. And we found that their responses during encoding also is kind of predictive of subject's subsequent memory performance. And next. And so for this data set, we have already put together as NWB files for each subject, including their behavior responses, the trial information, the task information, the TTL signals, the neural data like spiking times, and also some codes, which you can easily generate some raster plots directly from the NWB files. And next. So this is pretty short, and if you're interested, feel free to stop by at a poster session. Thank you.
All right, great, thank you. Diogo?
Sure. Good afternoon, everyone. I'm Diogo de Lucena. I'm a data scientist in the BCI team here at AE Studio, where we collaborate with research groups on software development to advance the state of the art of software and maximize the positive impacts of brain-computer interfaces. I'm here to present the data set, human FNIRS recordings of motor cortex during finger tapping task. Next. And this data set was actually collected by Dr. Rob Luke at Macquarie University, who is now part of AE as well, to answer the question if FNIRS can actually be used to classify different muscle actions, but with the main goal of having a high quality data set to be used for testing FNIRS softwares and methods. Next. And alongside him, Dr. Darian Slayton developed an extension for NWB data format to start supporting FNIRS data, which was used to convert this very own data set to NWB and then release it on DANDI. Next. So the experimental setup, what is in it? So the data set is composed by FNIRS data, next, of 5 participants, next, under 3 conditions, next. So either tapping the left thumb, tapping the right thumb, or no tapping for the control, with a block-based design 5 seconds long. Can you go next? And one of the main things here is that it's a very high quality data set, generally superior to more like traditional experiments, as it was really designed to be used for testing. Next. So what are the main findings? We have very high level of accuracy because of the cleanness of the data. Next. We demonstrated ability of transfer learning, so testing and training in one participant and testing in another one. Next. The data set has been used as a benchmark for software development, for instance, for FNIRS, for NIRS in the M&E package. Next. And it was successfully converted to NWB and released on DANDI using this extension that we created. Next. So how can it be useful? It's a very standard task and very clean, so very good for sanity check of software development. Next. Lots of trials and lots of short channels, good for testing algorithms and processing pipelines. You can sub-select trials and get a good trial versus accuracy curve, for instance. And it comes along with this MDX NIRS extension for NWB. So if you're working with NIRS data, please check it out. Next. That's all. Thank you, everyone, and feel free to reach out.
All right, great. Thank you. Colleen, you're up.
Okay. Oh, sorry. Okay, perfect. The first slide is there. Yeah, so I'll tell you guys about our OpenScope credit assignment project, DANDI set number 37. What we were interested in, as Jerome mentioned earlier, is understanding how predictive learning is occurring in the brain, and specifically how the somata and distal apical dendrites of pyramidal cells might be engaged in identifying the presence of unexpected stimuli in the visual environment. Next. So what we did is we initially habituated animals to expected stimuli, either a visual flow stimulus, as you see on the left, where the squares are moving, or a Gabor sequence stimulus, where you have these repeated A, B, C, D, G pattern sequences that occur one after the other. This was done without any imaging, and then we, during imaging, introduced rare unexpected events, so either sudden reversals of flow or unexpected sequences where the D image is replaced with a U. Next. And so what we were doing was 2-photon calcium imaging in primary visual cortex of mice that were on a running wheel in the original OpenScope setup. We were imaging in one of 4 planes, either a distal apical dendrite plane or a somatic plane in layer 2, 3, or layer 5 across several sessions. Next. As you can see, we're able to track both dendrites and somata. Next. So if you look at what we actually have in the data set, of course, we have ROI traces, we have tracking information, mask information. Next. We also have running velocity and pupil diameter for each of our sessions. Next. Quick glimpse of our results. We did find that both the distal apical dendrites and somata responded to our unexpected events, and they did so in different ways. Next. What you can see is that across sessions, we found that the distal apical dendrites were becoming more sensitive to the unexpected events, whereas the somata were losing sensitivity. Next. So that's just a quick glimpse of our results. Definitely check out our preprint, but also, as mentioned earlier, we've made a lot of resources available, from extended metadata to data analysis, and also stimulus code to reproduce our stimuli. So definitely do take a look, and definitely create an issue on GitHub or contact me if you're interested in using the data. Be happy to help. Thanks, Ben.
All right, great. Thank you, Colleen. Chris Rogers, you're up.
Hi, thanks for this time. Sorry I awkwardly popped on the screen during the panel discussion, but I'm grateful for the 2 minutes to tell you about my data set. I'm Chris Rogers. I'm a new professor at Emory University, and the data set I'm going to tell you about today was collected during my postdoc, and it is DANDI set 231. We were interested in one sentence in understanding how we can recognize the shapes in the world around us, and we used the rodent whisker system to analyze that. Next, please. And yeah, you can start the video. So I think these videos will use up most of my 2 minutes already, which is great. This is a head-fixed mouse who's interacting with these shapes. His job is to distinguish concave shapes like this one from convex shapes like you'll see on the next trial. While he does that, we track every contact that is made, every motion of the whisker in high speed and in great detail. So we've already done the pose tracking here to generate those little dots that are outlining the motion of each whisker. So I think the behavioral component of this data set is one of the things that makes it really exciting, the fine spatial and temporal detail that really tells us every single bit of sensory information that's coming to the mouse's brain as it makes this decision. And then can you go to the next slide? Thanks. And this shows us now simultaneous, you can go ahead and start the video, simultaneous behavior like what you've will be on the left, like the video you just saw. On the right will be large scale neural activity. So I recorded from many neurons across all the layers of S1 simultaneously. That's the red and blue hashes that you see there as spikes of excitatory and inhibitory neurons. There's a lot of detailed structure in this data. I've analyzed it kind of to a first pass in the paper that you can read about, but I think there's a lot more information out there that I hope some of you will be interested in uncovering based on the patterns of those that neural activity and how it relates to the behavior on the left. And if you could go ahead and go to the next slide. Right, this is the last slide. I've just had accepted a paper that really explains this DANDI set in great detail, including how it was formatted, how it was collected, all kinds of documentation about exactly what every field means. This is figure 1 from that paper, and it basically shows you the raw neural data, the spikes that I mentioned, the motion of the whiskers. That's already up on bioRxiv. You can check it out right now. It's very extensive documentation because I wanted to make it as easy as possible for anybody who's out there to be able to really dig into this data set and start making progress. And the NWB format was a really amazing resource for helping that to happen. And I think that's all I have to say.
All right, great. Thank you so much, Chris. Josh, you're up.
Hi, everyone. I'm Josh Siegel from the Allen Institute, and the data set I'm describing is from the Allen Brain Observatory called the Visual Coding Neuropixels dataset. And in this dataset, we have mice that are running on a wheel, but head fixed, and being passively presented with a wide array of both artificial and natural images. And during these experiments, we have 6 neuropixels probes inserted into various visual areas, and then the probes extend down into subcortical areas as well. Yeah, I mean, neuropixels have really become a new standard in electrophysiology because they have many more contacts and much denser contacts than competing types of electrodes. And so we wanted to take full advantage of neuropixels by sticking 6 into the brain at once. And as you can see on the right, in each experiment, we get spike trains from hundreds of neurons across up to 8 different visual areas, in addition to the regions that the probes pass through, such as hippocampus and subiculum. And so, yeah, these data sets, each recording session lasts about 2.5 hours, during which we show a range of visual stimuli, from receptive field mapping to drifting gratings to natural movies and natural images. And we have 2 different stimulus sets. One is called Brain Observatory 1.1, and the other is called Functional Connectivity. Each one is contained in a different DANDI set, either 21 or 22. And then we also have recordings from mice of different genotypes expressing channel rhodopsin in either parvalbumin and somatostatin or VIP positive interneurons in cortex. And at the end of each recording, we do a brief optotagging session to identify cells of those particular types in the recording. So in total, we have 58 recording sessions packaged in NWB format. In the lower right, you can see the breakdown of genotypes by the different stimulus sets. And this data set is highly complementary to the visual coding 2-photon imaging data set, which I think Saskia will talk about in a second, and is also, is there a second slide here? Oh, I thought I only had one slide, but okay, yeah. So this slide shows you the breakdown of the total number of units across all the different areas that we've recorded, as well as some of the example use cases. So there have been a number of papers by people external to the Allen Institute that have used this data set for novel discoveries. It's also common to use it for validating new analysis methods. And then another use case that we really like is when people directly compare data collected externally to the data we've collected and look for similarities and differences. So I think there's still a lot of rich information in this in these data sets that haven't been looked at, a lot of new dimensions for potential analyses. So happy to talk to you during the poster session to give more info about what's included and how you might be able to apply it to your science.
All right, awesome. Thanks, Josh. Next up is Felix. And Felix submitted a video, so here you go.
Hello, I'm Felix Pei, a student in Chetan Pendarnath's lab at Georgia Tech. I'll be talking briefly about the DANDI sets in our 2021 Neural Latence Benchmark Suite. The Neural Latence Benchmark is an effort to standardize evaluation of latent variable models of neural population spiking activity. We use preprocessed dataset sessions from 4 different experiments for the benchmark. The datasets all include sorted unit spiking activity recorded from different regions of the macaque brain in various behavioral settings. The first dataset is the MC Maze dataset from the Shenoy lab at Stanford. Activity was recorded from the motor and premotor cortices during a delayed reaching task. The dataset has been used for studying dynamics of movement preparation and execution. The next dataset is MCRTT from Joey O'Doherty in the Sabies lab at UCSF. Activity was recorded from the motor cortex during a random target reaching task, which provides a more realistic setting for examining neural activity during naturalistic movements. The Area 2 bump dataset is from the Miller lab at Northwestern. Activity was recorded from the somatosensory cortex during a reaching task where, on some trials, the subject's arm was perturbed before the reach. The dataset offers a window into how active and passive arm movement is encoded in sensory regions of the brain. Lastly, the DMFC RSG dataset is from the Jazayeri lab at MIT. Activity was recorded from the dorsomedial frontal cortex during a cognitive timing task, and the dataset has been used in research showing how neural dynamics during the task correspond with Bayesian inference. If you'd like to learn more about the datasets in our benchmark suite, please come by during the poster session.
Okay. Whoops.
Hello.
Move forward. There we go. All right, Satya, you're up.
Great. So I will tell you about DANDI set number 39, which is about contrast tuning in the massive visual cortex that was collected with 2-photon calcium imaging. So advance, please. So this data set leverages the standardized data collection and processing pipelines for the Allen Brain Observatory, which is our high throughput data collection pipeline. And so you can see we use transgenic calcium indicators to record activity in the very dim movie that's playing on the right, if people are able to see that next. So for this, this data set, is we looked at contrast tuning in the visual cortex. The motivation is kind of multifaceted, in large part. So contrast tuning is something that's been studied psychometrically for a long time, as well as physiologically, and we wanted to look at how this was organized in the visual cortex. So we used a drifting grading at 6 different contrasts and moving in 8 different directions. And there's more details about the stimulus here. And this allows us to look at the contrast tuning of individual cells and see how this differs across layers or differs across areas within the cortex. Next. So we imaged in 6 different cortical areas. V1 is labeled here as VisP, and then there's 5 higher visual areas that surround it. And we used transgenic lines that limited expression in specific cell populations. And so these included 4 excitatory lines that were layer specific. So we're able to image in layer 2, 3, layer 4, layer 5, and layer 6 in the excitatory pyramidal cells. And then we used 2 inhibitory lines to target the VIP and somatostatin inhibitory interneuron populations. Next. We didn't sample all of these combinations, all of the areas and transgenic lines completely. So you can see this chart kind of shows you where data was collected. So for instance, layer 2, 3 was imaged across all 6 visual areas that we collected data from, whereas some of the other lines were only imaged within V1. I want to point out that the paper that we've published with this data set actually only analyzed the data from V1. And so there's a lot of work that can be done looking at the contrast tuning in other visual areas in this data set. Next. So just really briefly, I'll tell you, we have analyzed this data, the V1 data, and have a paper next that shows looking at the contrast tuning of different cells, and we find some cells that are high contrast preferring. So as the strength of the stimulus gets larger, the response of the cell increases. Other cells are low contrast preferring. Next. And this, we see kind of differences in the distribution of the low contrast and high contrast preferring cells across the different types of cells. So for instance, the VIP interneurons are almost exclusively low contrast preferring, whereas the somatostatin interneurons are almost exclusively high contrast preferring. And then as we move down the layers of the cortex, we see an increase in the high contrast preferring, the number of high contrast preferring cells. Next, we did some modeling, next again, sorry. Did a stabilized superlinear network model, and we're able to kind of demonstrate the involvement of the VIP and somatostatin interneurons in regulating the dynamic gain of the cortical circuit. And I can tell you more about this if you're interested. Next. So there's some open questions, I think, with this data set. As I mentioned, we haven't really analyzed the data across the cortical areas. So I think there's a lot of work that can be done looking at contrast tuning in the higher visual areas. I think there's a lot of questions about these low contrast preferring neurons that are found in layer 2-3, especially in the pyramidal populations, and what they might be doing and whether they're also present in the higher visual areas. I think another question that I forgot to add on here that would be really interesting to look at in this data set is how the population statistics might change with contrast, as we have large populations of neurons in each recording. So I will stop now, but do come to the poster. I'm happy to tell you more about this data set.
All right, great. Thank you, Saskia. And Simon, you're up.
Hi, everybody. Yeah, my name is Simon. I'm from the Fleischmann Lab at Brown University, and we are broadly interested in outdoor information processing in the mouse olfactory cortex. Next. Next, please. Yeah. And so we provide this DANDI set number 167 in order to tackle the main question as how is information that is computed by the olfactory cortex that is broadcasted to downstream brain targets. Next. So the olfactory cortex is the main recipient of olfactory bulb input, and it's thought to encode odorant information through population coding in high dimensional space. Next. That piece of brain also projects to several downstream brain targets. Next. And so what we hypothesize is that these piriform projection ensembles will differentially compute sensory information. Next, and our approach was to record the neuronal activity of two different projection ensembles in naive animals. Next. In order to do so, we use 2-photon calcium imaging in head-fixed animals running on a wheel using microendoscopy using a green lens, and we presented a panel of 10 different odorants, 8 trials each. Next. And we recorded through multiplane imaging the activity of several hundreds of neurons, next, as well as behavioral information as sniff and locomotion. Next, next. And after segmenting the data, next, we looked at the odor responses of all of our cells that we were able to record. Next, and looking at population coding, we specifically looked at 2 different projection populations. Next, and at the bottom is our main findings, where we saw using a linear classifier trying to decode the identity of the stimuli on each trials, that the odor productivity of those two different samples in magenta and cyan were actually different, giving us a hint as how the information from the olfactory cortex is broadcasted to downstream brain targets. Next. And so in summary, what we are giving as a DANDI set here is a recording of olfactory cortex neurons in awakes and head-fixed animals during passive odor presentation experiments, including some behavioral data as of sniff and the distance of the mouse walking. This dataset contains data from 5 mice, about 1,000 of neurons, where a panel of 10 different stimuli, 8 trials each, were presented. And next. And all of this is in the NWB formats, and we are also providing a pipeline in order to convert the segmenting data that we are using with Suite2P into NWB, as well as a notebook in order to easily open that data set and start to play with it and analyze the data. Next, and I will just mention that that dataset contains a second non-functional red channel that is currently not available in order to tag those particular population of neurons where they project to, and that will be available later this year. And if you want to, and if you have any question, I'll be happy to chat with you during the session right after. And you also have our email address at the bottom. Thank you.
All right, thank you so much, Simon. And thanks to everyone. That was the last one. So now it's time for the poster session on Gather. So if you are interested in, particularly interested in any of those, definitely go to the corresponding poster on Gather. So I'm gonna stop my share and make sure that we have, yeah, okay, so Carly just put the link in there. So we're all gonna go over to Gather. I'm gonna stay on here for a minute just to answer any technical problems any people might have, but go on and head over, including the presenters and all of the attendees.